Should we start, Professor Ganguly? Good afternoon and a very warm welcome. Respected Chief Guest, Professor G.N. Kazi. Respected Guest of Honor, Professor Yoshifumi Takeda. Our esteemed keynote speakers, chairpersons, presenters, delegates, senior colleagues from the Yakult Central Institute for Microbiological Research and Denon Research Center, and our young investigators. We would really like to thank you for making the time for participating in this initiative of the Yakult India Microbiota and Probiotic Science Foundation. This foundation is an initiative of the Global Probiotic Council that was established in 2004. The primary objective of the foundation, in keeping with the aims of the Global Probiotic Council, is to promote and advance the science of the gut microbiota and probiotics. The foundation was registered as a society under the presidentship of Professor Nirmal Kumar Ganguly on 9th November 2011 under the Societies Registration Act 21 of 1860. As the foundation forges ahead to capture the renaissance in therapeutic microbiology and takes the science of the gut microbiota and probiotics to the next level, it gives us immense pleasure to welcome you all for this series symposium, which is being organized by the foundation in association with the Translational Health Science and Technology Institute and the Apollo Hospitals Group. The program this year has especially been designed to unravel the factors that shape the microbial communities of the gut and identify the latest developments in probiotic science that will positively affect the microbiota for clinical intervention. Recognizing the need to promote probiotic research in the country, the foundation has instituted 3 Young Investigator Awards to felicitate talent. The response for the announcement of this award was overwhelming. In fact, we received 62 applications. And after a stringent evaluation by the governing body of the foundation, we are pleased to welcome 20 young investigators who will be presenting their posters, which will be evaluated for the first 3 positions by a jury comprising of eminent national and international scientists. I think the young investigators deserve a round of applause. The presentations of the symposium will also be webcast, and the link of the webcast will be sent to all the delegates so that you can have an access to the presentations should you wish to revisit them. The proceedings of the symposium will be edited by international and national experts, Professor Yoshifumi Takeda, Professor Balkrishna Iyer, and Professor B.S. Ramakrishna, and will be published as a book with an ISBN number. The book will be distributed to healthcare professionals across the country and to libraries in the country to serve as a repository of information. We would once again like to thank each one of you and all of you for having taken the time to attend this symposium, and we hope you enjoy the presentations and deliberations that ensue over the next 2 days. We will now proceed for the lamp lighting ceremony, which is a typical tradition in India before the commencement of any important event. We are honored to have among us Professor G.N. Kazi as the chief guest of the symposium, Professor Yoshifumi Takeda as our guest of honor. I would like to invite Professor G.N. Kazi, Professor Yoshifumi Takeda, Professor Nirmal Kumar Ganguly, Professor G. Balkrishna Iyer, and Professor Anupam Sibal to please ignite the lamp. I now request our esteemed dignitaries to kindly be seated on the stage. May I now request Professor Nirmal Kumar Ganguly, the President of the Foundation, to kindly deliver the welcome address. Professor Nirmal Kumar Ganguly needs no introduction. He was the former Director General, Indian Council of Medical Research. He is now a visiting professor of eminence in the Translational Health Science and Technology Institute, Gurgaon, President, Asian Institute of Public Health, Orissa, President, Jawaharlal Institute of Postgraduate Medical Education and Research, Pondicherry. Professor Ganguly has several national and international fellowships and has received more than 100 national and international awards, including the prestigious Padma Bhushan awarded by Her Excellency in the field of medicine in 2008. He has more than 750 publications in peer-reviewed journals and 22 book chapters. Professor Ganguly, please.
Professor G.N. Kazi, Professor Yoshifumi Takeda, Dr. Sebbal, Dr. Balkrishna, all the dignitaries who have come from abroad, and our own colleagues and friends in the audience. I will be very happy to. Nirja told us that we established ourselves as a foundation in 2011. But a strong group of academicians had started it with the help of Professor Takeda, the first meeting we had done in the NICED. And our sole donors at that time were the Yakult, who had come in India at that time, and they kindly did it. And we did it so successfully that at later periods we went on doing it under the aegis of NICED and ICMR. And at one time we realized that we should have our own existence. And this group, which were many of us are here today, Dr. Nayak, Dr. Mittal, Dr. Ramakrishna, Dr. Bhatnagar, Dr. K. Lahiri, I could go on taking name of Dr. Sasikaran. All of these guys, they got together and thought that we will start a foundation. It was very easy. We were all academicians. It was very easy to start a foundation, but we didn't know from where the money is going to come. And so we then approached our original funder, and very kindly they agreed to fund us for the next 5 years. And but even when we got started, we found out that even if you have a funder, it is very difficult to take money. I thought that it must be very easy to take money, but to my horror, we saw that it was not very easy to take money. So we took a loan, and this loan was very graciously given. And we ran the foundation on loan for the last couple of years. But today you will be very happy to know that we have been able to receive the money and the loan has been paid off. So we are now free of loan burden, and we are real foundation. And thanks to our donors that they, without any strings attached, allowed the foundation to function as a foundation and an academic organization promoting the science of probiotics. Today, for a couple of minutes, I will bring in few things which is also theme of this particular meeting, one of the themes of this particular meeting, how the regulators will look at probiotics when lot of things come in people's mind. Like one of the things which often come in people's mind, that this is a food which has organisms. So if it has organisms, whether bacteria and yeast, how this could be a healthy food. So this particular thing is even in regulators' mind. So we will like to tell that there is a lot of research which has gone on, and lot of traditional memories which have gone, knowledge which has gone on, that when we are born, when we are in mother's womb as a fetus, now we know that we might have some bacteria, but we are really in a very sterile environment. Science says now that is not that sterile, but we will like to believe we are in sterile environment. And when we are born through natural processes, we acquire certain healthy bugs from our mother, which populates us, and that allows us to live. If we didn't have that, we would have died. Those babies who are not breastfed, we learnt it when we were saving babies from AIDS by treating the mothers and babies. We found that we didn't allow them to be breastfed because we thought that they will get the virus. That policy has been changed now. So we saved the babies, but the babies died of lower respiratory tract infection and bloody diarrhea. So it was futile. So this shows that we need to have bugs to populate our mucosal system, to build us our immune system. And all of us know that our immune system is built up through the gut. And because the gut is the primordial organ, we have, of course, we have thymus. But if you go to the chicken and birds, you know what Bursa fabricius does. So, and you take any food, right from the curd to your bread, loaf of bread, to some cured meats, to cheese, to your curd, even to dosa and idlis, all of them have bugs, and nice bugs with whom we are familiar. So we should be able to look at the real science which goes behind this. There are a series of bugs which are very friendly to our body. And if you go to the posters now, you look at that the bugs which are unfriendly bugs, they are in the colon cancer, they are in ulcerative colitis, they are in diabetes. Whenever we have a disease, we have unfriendly bugs, and when we are healthy, we have good bugs. And the new science of metagenomic has proven this without any doubt. And some of the major work has happened in India around Dr. Ramkrishna, Dr. Balkrishna, and some of the youngsters who are sitting here. So this is again the science which shows that healthy bugs are necessary, and these healthy bugs need to be obtained in food from time to time, whether it is breast milk, whether it is coming from the food, whether it is coming in a fortified system, it is needed. So there's one thing which worries the regulators, worries the general public, and I try to explain this in the scientific terminologies. The second thing which worries people, because this is one major worry, that when we say that it builds up your health, and when we say that it builds up your immune system, do we have evidence? This is one of the things where there is no doubt. One of the greatest immunologists actually was the voter of the probiotic, Dr. Eli Mechnikov, if all of us remember. He was one of the proponents of the innate immunity, and he was one of the biggest votary of this. And we now have innumerous publications showing how the T cell expansion, T cell regulation, toll receptor related regulation, all of these things are gradually built up to provide us a healthy immune system and abrogating the bad immune system. Sharat Gopalan is here. He was telling me this afternoon that after diarrhea, one of the major publications in probiotic is coming up in the atopic allergies and atopic systems, and this is one of the major published area. And you will be very happy to know that when I went to the net, I found out this is one of the largest published area in a one single domain in the scientific literature at this moment. It's mind-boggling. So this particular thing goes with the probiotic as its generic power of building up the immune system. And we know that immune system is one of the host parasite system which controls paracrines, exocrines, mucosal secretion, mucosal health. Almost everything in our body is controlled by the immune system. So, if one could say that whether probiotic food builds up health, you don't need to go any further. There is a poster also there today. Right? It was an elegant poster which takes up real genomic science and other science to tell us that the probiotic is probiotic in food is also builds up health. Another thing which might worry people, and this is something which is worth worrying, that how probiotic, because when you go to the poster, sometimes you will start asking that all the posters which are there, they show that somehow or other probiotics have helped in one disease or the other. How it gets done? Today, one of the major research area is not a disease-oriented area. Major research area is how the inflammasomes work. And if you see, there are huge number of papers in the journals Science, Nature, and Cell on the inflammasomes. And you take a disease, it is an inflammatory disease, right from coronary artery disease to diabetes, even to obesity, the infectious diseases. You just name one, and cancers. Inflammation is central to all these things, and it regulates our metabolic system in a fashion which is deleterious. So anything which works on inflammation is very, very healthy, and this is something which probiotics does. It is one of the very nice anti-inflammatory system in our body. So again, this is another very major evidence that it keeps us healthy if we have our gut populated by appropriate organism. The final point which I will like to make here today, that this science is very rapidly evolving. There are two kinds in the probiotic setting. One is that there is a specific health claim, that it works on pouchitis, it works on mucositis, it works on maybe gluten allergy, reducing the after effect of gluten allergy in celiac disease. There you need evidence of clinical trials done in a very sophisticated fashion, and you need strong evidences on not only one trial but several representative trials around the globe, and doing appropriate meta-analysis and establishing that it works. But when it is about general health, when it is about building immune system, it is a different story. And I wanted this differentiation to be known here because, and those who are working on nutrition, you sometimes say to your guys that milk is healthy or carrot is healthy or take papaya in the morning, as Dr. Talwar perhaps does. Welcome, Dr. Talwar. He is one of the major researcher in the probiotic. So these are traditional knowledge, and some of the knowledge has come out of the research. But hardly anyone does a clinical trial with that. Trials have been done with overall food, but hardly one does with a single vegetable and others a clinical trial. But these have been established with gathered knowledge which has been managed and which has been analyzed properly to put it in an appropriate place. And I think any food or any product should carry that particular burden, but not always a strong clinical trial to prove the point. But if it is a specific health claim, it has to be done. Now, one thing which most probiotic foods suffer from is having some food additives which people could worry about. One of the substances is sugar. Around 10% sugar might be a part of the probiotic product. As you know, that huge number of food, some of them are non-nutritious. Mind you, the caramelized carbonated drinks are non-nutritious, but it is in every home. Dr. Sunita Mittal's house also have this, and this particular product is non-nutritious. It has 12% sugar, and this sugar is also not, people say that it is not desirable, but only if you take 4 or 5 bottles of this, then it is not desirable because you are taking too much sugar. But every one of us here, sitting here, takes a cup of tea, and when I push them aspartame or sucralose, they say, no, let us have at least our spoon of sugar. And that is about 5 grams of sugar. And it doesn't do that much harm unless medically you are not allowed, or you have to reduce sugar. In this, luckily, the most of these don't have preservatives which harms us, or excipients which harms us. So it is worth worrying, worth creating a product line which might help those who can't have probiotics with some of the ingredients. But overall, for the general public, it really doesn't matter, and it should not be a major health concern, because a cup of probiotic food or a small vial of probiotic food doesn't carry that much substance, and it is never taken that you go on taking it as you are taking a plate full of rice. It is taken in small quantities. So these were some of the concerns which concerned us and which was asked from me by both regulators as well as general public as well as scientists. So each one of the statements which I have made here are supported by a huge amount of scientific data and publications, and I tried to use a very general terminologies and a simple language to explain it to this audience, which perhaps was thinking that what's happening, because all of you are scientists, and when I look around the room, this audience I'm very happy to have. This has got pediatricians, gastroenterologists, the medical scientist, the nutritionist, the scientists from the Lady Irwin College, very hardcore science people from THSTI, the scientists working on the different research departments, oncologists, immunologists. Almost everyone of the stakeholders are sitting here and rubbing shoulder, and perhaps at the end of the day, we'll learn from each other. Sorry, Nirja, that I took more time than allotted to me, but I thought that because the regulation was one of the part, and you have not allowed me any time, or you never invited me to give a lecture, I thought I gave a lecture on this particular occasion. Thank you.
Thank you, Professor Ganguly. That was a very enlightening address, and next time we'll have a special lecture only by Professor Ganguly for all of us. I would now like to invite Professor G. Balkrishna Nair, Executive Director of Translational Health Science and Technology Institute, Gurgaon, Vice President of the Foundation, and the Principal Organizer of this symposium to deliver the welcome address. Professor Nair is an internationally renowned scientist and a fellow of several national and international academies. Among others, he was awarded the prestigious Shanti Swarup Bhatnagar Award for Medical Sciences in 1998 for his contributions to the discovery of Vibrio cholerae O139 Bengal. He has more than 490 publications in peer-reviewed journals. We would like to thank Professor Nair and THSTI for the tremendous support in organizing this symposium along with the Foundation. Professor Nair.
Distinguished Professor Kazi, Professor Nirmal Ganguly, Professor Takida, and Professor Sibal, and distinguished members in the audience. First, I would like to start by saying it's a big pleasure to co-host this symposium along with the Foundation and along with the Apollo Hospitals. It's a great privilege, and thank you for giving us this privilege. And second, on behalf of THSTI, all its staff, we would like to extend a very warm welcome to the symposium. I'm sure at the end of tomorrow, or at least by the afternoon of tomorrow, you will be educated a little bit more than what you've come here with. But what I've done is I've chosen this time just to show you 5 slides about what THSTI is. THSTI is new in 2 areas. As an institute, it's new, and as what it does, it's new. Because if you ask 10 people what is translational research, you'll get 10 different answers. But more and more, it's now coming to light that translation in health is just moving knowledge from basic research to clinical applications. If you remember that, I think that's the most elegant definition of what translation is. And that's what this institute, which we abbreviate as THSTI, acronym as THSTI, and of course it's known as the Translational Health Science and Technology Institute. It's a new institute funded by the DBT or under the DBT and the Ministry of Science and Technology. So just some simple questions of who are we and what do we do? The institute was established in 2009. It's an autonomous institute under the Department of Biotechnology, as I said, and under the Ministry of Science and Technology, Government of India. It's an institute which grows on an institutional ecosystem of multidisciplinary research for translational research to innovative for improvement of public health. To innovate, and its moving knowledge, as I said, came from basic research to clinical applications. This is the mission of the center. It's by integrating the fields of medicine, science, engineering, and technology into translational knowledge and making the resulting biomedical innovations accessible to public health to improve the health of the most disadvantaged people in India and throughout the world. That's the mission of the center. The organogram looks something like this. It's got niche centers, or what we call as intramural centers, 6 of them. You have the, to listen, I won't read them out, it's there on the slide. Then we have the extramural centers, which has which has 2 components to it, and the partnership centers. Now, a lot of people tend to tell me that, how are you different? You know, you have named, instead of departments, you have named them as centers. But the difference actually is the way we work. And the way we work is not horizontally, not each center going into doing work of its own, but vertically, with centers amalgamating with each other. Say, for example, we have in the pediatric biology unit a program known as preterm birth or biology of birth. But that's not only done by pediatric biology unit center. It's done by multi-centers where the microbiome chips in, proteomics chip in, metabolomics chip in. And so it's a program-based research institute which doesn't subscribe, individuals are not doing their own research. But the concept is to get the best of the program from all the kinds of expertise that's available within the institute and outside the institute, very open. One of the centers there, known as the Center of Human Microbial Ecology, is one which delves into the microbiome. And there are 2 microbiomes which we are focusing on. One is the gut microbiome and the other is the vaginal microbiome. Studies along those areas have started, and that's why this foundation means a lot to us. That's how the institute would look. It's an artist's conception. You could, on my left is the Regional Center for Biotechnology, which is there, and the Translational Health Science and Technology Institute. It's kind of amalgamation of institutes, and we call it the NCR, or the National Capital Region Biotech Bioscience Cluster. And this is expanding. This is what the artist's conception is, but maybe in the course of this year, we'll shift into this institute, which is almost up and about. So, again, from the Translation Health Science and Technology Institute, a very warm welcome to this symposium, and we look forward to excellent interactions. Thank you for coming.
Thank you, Professor Nair, for an insight into Translational Health Science Technology Institute. And we are privileged at the association because I think probiotic science is all about translation. And now I have the privilege of inviting Professor Anupam Sibal, the Group Medical Director of Apollo Hospitals Group and the Principal Organizer of this symposium, to deliver the welcome address. Professor Anupam Sibal is a Senior Consultant Pediatric Gastroenterologist and Hepatologist, Indraprastha Apollo Hospitals. He is a Fellow of several national and international academies. He is a recipient of several awards and has more than 85 publications, including book chapters. The Foundation would like to thank Apollo Hospitals, particularly Professor Anupam Sibal and Dr. Sarath Gopalan, who have given all the support in organizing the symposium. Thank you so much.
Thank you, Nirja. Very good afternoon, Professor Kazi, Professor Takira, Professor Nirmal Ganguly, Professor Nair. The galaxy of experts that I can see in this hall, and it's a really distinguished gathering. Professor Santosh Mittal, Professor Talwar, Professor Sasikaran, Professor Philip, Professor Shinjini Bhatnagar, Professor Rajiv Bhavdekar, Ashish Bhavdekar, Rajiv Redkar, from the field of dietetics, Dr. Puri, Dr. Kumud Khanna, Dr. Rekha Sharma, Dr. Bhuvaneshwari, and I could keep going on and on. This is indeed a spectacular gathering. It's a very hard act to follow Professor Ganguly, because he doesn't update his knowledge on a monthly basis, fortnightly basis, weekly basis, or a daily basis. He does this on an hourly basis. So I won't even try and actually get into the science of probiotics. And thank you, Professor Ganguly, for wearing that lovely red muffler on this cold, by Delhi standards, February afternoon. I read this yesterday and I want to share this with you. An Alu went to a Bhindi and said, I want to be friends with you. And the Bhindi said, I am so slim, I am so pretty, look at you, you're so fat, you're so round, I don't want to be friends with you. So the Alu said, fine, never mind, I'll make other friends. And today we have Aloo Gobi, we have Aloo Matar, we have Aloo Gajar, but you only have Bhindi. And what I'm trying to say here is that everyone needs friends. And what this meeting, I think this symposium makes it unique is the fact that you have not limited yourselves to just clinicians, which is the case with 90% of the meetings in India, where you just have allopathic doctors just talk about stuff. But here you have nutritionists, you have dietitians, you have basic researchers, you have microbiologists, you have basic scientists, you have adult gastroenterologists, pediatric gastroenterologists, pediatric surgeons, pediatricians, and the list is just so vast. And I think we need to congratulate the foundation for bringing together such an interesting, such a diverse group of experts, because it's only through friendships and collaborations that we learn. So I'd like to congratulate you for your initiative in doing this. This morning I Googled probiotics and I got 5 million results in 0.18 seconds. And then I went on to PubMed and I found 10,406 peer-reviewed papers on probiotics. So clearly, there is a lot of interest in probiotics, there's a lot of research going on, and the interest seems to be growing every day. And when you have as many as 10,000 peer-reviewed papers, then there clearly is an opportunity for conferences like this where you can actually make sense of that research, because not everyone is as accomplished as some of the experts here to be able to make sense of the huge sea of knowledge that exists and is available. And for those of us in clinical medicine, this becomes even more important, because when we see patients, the families today have researched a lot of things when they come to us, and therefore for us to be able to make a useful or a scientific decision, we really do need meetings like this that actually look at evidence. Now, of course, for years there's been a lot of interest in the gut and probiotics, starting with antibiotic-associated diarrhea, C. difficile, then of course interest in viral diarrhea, traveler's diarrhea, inflammatory bowel disease, irritable bowel syndrome. But now, of course, there's a lot of interest outside the gut. If we look at the oral cavity, there's interest in preventing tooth decay, in gum disease, the skin, atopic dermatitis, respiratory allergy, autoimmune disorders such as rheumatoid arthritis, fatty livers, vaginitis, and the list goes on and on. And while we were really looking at a handful of agents, the list of agents that are being studied has also increased considerably over a period of time. In February this year, there was a paper published in the Proceedings of the Nutrition Society, and it actually talked about how by modifying gut microbiota, one can actually alter the risk of cardiovascular disease. So what was once believed to be true, and we all believe that a way to a man's heart is through his stomach, actually a way to a healthy heart might be through healthy microbiota, and maybe 5 years from now, we're actually going to be debating more and more of that. From the Apollo Hospital's point of view, it's a privilege to co-host this. We consider it a privilege to have been asked by the foundation to partner with such a distinguished institution such as Professor Nayar's. And while we are largely recognized as Asia's largest integrated healthcare delivery system with a presence in primary, secondary, tertiary, clinical care, health, IT, informatics, pharmacies and insurance, over the last 5 years, under the leadership of Professor Ganguly, Professor Roy Chaudhary and Professor Sita Naik, we've actually laid out a very strong foundation in the area of academics and research under the Apollo Hospitals Education Research Foundation. We now have 600 postgraduate trainees in 35 DNB programs. Our first medical college opened in Hyderabad 2 years ago, and we have been involved in some very serious research efforts, including defining the normative value for the Indian population based on a study of 80,000 Indians who came to our clinics for preventive health checks, where we found nothing wrong on history or examination. There's some interest in heart disease. We are looking at the genetic predisposition to heart disease. We're looking at utilization of ECGs for predicting much more than what ECGs usually predict. Some other work in the area of finding out who's going to respond to antiplatelet drugs, and the list is pretty long, and I think we, as part of AHERF, consider it an honor to have been asked to co-host this conference, and we would welcome such opportunities in the future. I think this is going to be an amazing conference. We don't have any empty chairs, Nija, so well done, and congratulations to everyone at the Foundation for bringing all of us together. Thank you very much.
Thank you, Professor Sybil. That was a very interesting address. And in fact, it is the privilege of the Foundation to be associated with Apollo Hospitals Group and Professor Sybil and Dr. Gopalan and all the esteemed delegates. We are honored, privileged, and overwhelmed to have with us Professor Yoshifumi Takeda, who held the torch many years back and showed us the way. We are here because of you, Takeda-san. Thank you so much for your esteemed participation. Professor Yoshifumi Takeda graduated from the School of Medicine, Osaka University in 1960, and he initiated his scientific career in 1961 as a student of Dr. T. Fujino, the discoverer of Vibrio parahemolyticus at the Research Institute for Microbial Diseases, Osaka University. During the past 50 years, he has been working on the pathogenesis of several enteric bacteria, such as Vibrio parahemolyticus, Diarrhegenic E. coli, and Vibrio cholerae. The role of toxins produced by enteric bacteria has been the focus of his research interest. Among his many contributions, the outstanding ones are the demonstration of the cardiotoxicity of the thermostable direct hemolysis produced by Vibrio parahemolyticus, study on the structure-activity relationship of the heat-stable enterotoxins produced by enterotoxigenic E. coli, demonstration of the RNA N-glycosidase activity of Shiga and Shiga-like toxins produced by Shigella dysentery and enterohemogenic E. coli, and discovery of the new cholera toxin-producing serogroup known as Vibrio cholerae O139 Bengal. Professor Yoshifumi Takeda has worked as professor and chairman, Department of Bacterial Infections at the Institute of Medical Sciences, University of Tokyo from 1983 to 1989. Professor and chairman of Department of Microbiology, Faculty of Medicine of Kyoto University from 1987 to 1995. Director General of the Research Institute of International Medical Center of Japan from 1994 to 1999, and Director General of the National Institute of Infectious Diseases Japan from 1999 to 2001. From June 2007 to November 2011, he worked as the Director of Collaborating Research Center of Okayama University for Infectious Diseases in India, which is located at the National Institute of Cholera and Enteric Diseases, Kolkata. He is currently an emeritus scientist at the National Institute of Infectious Diseases, Tokyo, Japan, and is also the president of the Hideo Noguchi Foundation. His current research interest is on viable but non-culturable Vibrio cholerae, on the newly emerged Vibrio cholerae L-TOR variant, and on a heat-killed vaccine against the disease known as shigellosis or bacillary dysentery. Professor Takeda was the leader of the team that conducted the largest community-based field trial in Kolkata to evaluate the impact of the probiotic bacteria, Lactobacillus casei strain Shirota, on prevention of diarrhea among children in an urban slum in Calcutta. He has also made significant contributions in understanding the gut microbiota of impoverished children and adults. Professor Takeda was responsible for creating the unique JICA Center of the National Institute of Cholera and Enteric Diseases in Kolkata. In recognition of his outstanding contributions in enteric diseases, in recognition of his mentorship to a host of students from India and other Southeast Asian countries, and in recognition of his love for the people of India, the Yakult India Microbiota and Probiotic Science Foundation is privileged to bestow on Professor Yoshifumi Takeda the Lifetime Achievement Award. I would now like to request Professor Nirmal Kumar Ganguly and Professor G. Balkrishna Nair to please give Professor Yoshifumi Takeda the Lifetime Achievement Award.
It has been a big honor and privilege to shake hands with Dr. Takeda. Myself and Dr. Balkrishna Nair has been his closest associate when I was working in Japan in the same institute as he was working. And Professor Balkrishna Nair started his career as student of Tai Takeda, late Dr. Tai Takeda, the wife of Dr. Yoshifumi Takeda. And since then he has been constantly associated with him and was a co-researcher with him. Thank you, Professor Takeda, for this privilege.
It's really my great honor and pleasure to have received such a prestigious award. Thank you to the foundation as well as Dr. Ganguly and Dr. Nair for their selecting me to receive such a wonderful award. Being sitting here on the podium, I remind of the day, a very hot summer day in 2007, when Dr. Ganguly, Dr. Nair and myself have discussed about a possibility to have a symposium for probiotics in India. At that time, I was based in Calcutta at National Institute of Cholera and Enteric Diseases, directed by Dr. Nair. A baby symposium we raised at that time has grown up as you see today. I'm very happy to see. Having received such an award, I have decided to work harder to make this symposium more and more successful. Thank you very much.
Thank you, Takeda-san, and congratulations. We are privileged and honored to have you with us today. We now move on to the inaugural address. We are honored to have with us Professor G.N. Kazi, Vice Chancellor, Jamia Hamdard University, as the chief guest for this symposium. May I request Professor Nirmal Kumar Ganguly to kindly introduce our chief guest.
Actually, this was not meant like that, but today in the dais, it's a kind of a friends association. Because each one of us have worked with each other for a very, very longish time period. So Dr. G.N. Kazi I know him for decades now, and he has been the director of the IIIM, one of the institutes in India which does research in traditional medicine, and during his leadership in the modern biology, modern science, and try to give a traditional medicine a new twist. Some of his biggest achievement, mind you, he was an enzymologist, a biotechnologist, but he transformed himself admirably while leading that institute to make it one of the most modern institutes of drug discovery, drug research, as well as establishing Indian system of medicines into a stronger platform. And his contributions were that our total knowledge was unprotected. It was not covered by any documentation, and he created that documentation in the CSIR and put that on the forefront. He now chairs some of the very vital committees in Government of India, and also helps some of the other nations to understand the knowledge which flows through in this particular science. And with his leadership, we are getting more and more known globally in the area of our traditional knowledge, and linking it to the modern medical knowledge, as well as in the areas such as nutraceuticals and substances which help us. Another very major area where he worked, and which is also very, very relevant in today's context, that he created tremendous knowledge for Indian farming community, and particularly the farming community in Kashmir Valley. And it was a real privilege to work at that time with him there, when all around us the environment was slightly warmer in Srinagar, but he didn't care about anything, and all around him prosperity flowed because he contributed tremendously in that. Recently I have started another stint with Dr. Qazi. When he became the Vice Chancellor of Jamia Hamdard University, he modernized that university and only uttered 3 words to everyone: research, research and research. So people sometimes tell me that your friend only talks about research. I said, yes, that is what he was doing all his life. So he talks about research. And this is in his core of his heart. He is in his heart, he is a hardcore researcher, and he strongly believes, and I also believe in the same thing, that research is one of the major catalysts for academics and knowledge. And the new thing which he is building, and with amazement I was seeing that, I thought, from where will he get money and from where he will get faculty. He is building a first-rate medical school and a very strong super speciality structure over there, and like a wizard has been able to generate funds and financing to fuel that enterprise and move that forward. It is coming at one of the amazing institute around him, and another feather to his cap. I'm sorry I didn't read what is written there, but gave you a different kind of introduction. So it's privilege, Dr. Qazi, that you could find time from your very busy schedule to come here.
Very good afternoon, ladies and gentlemen. Professor Ganguly, Professor Yoshifumi Takeda, Dr. Balkrishna Nair, Professor G.P. Talwar, Professor Sasikaran, Professor Anupam Sibbal, Dr. Neerja Hajela, and many, many other distinguished invitees here, and speakers and students. It's indeed a great pleasure for me to be here this afternoon. I fully agree with Nirmal Ganguly when he said that he should have been called for a formal lecture, but by saying whatever he did, he has already bored me because I will not say anything more than what he said. Let me start first of all by saying that I am privileged to be invited to this symposium, and I will take another maybe 25 minutes to say what most of you already know, and what I would just like to start like a starter. You know, when we make yogurt, we just put some starter in that. That's my role today. Not that I can update your what is happening in the area of probiotics, but I will just try to bring in some of the things which are very well known. And by the way, Neerja flooded me with a lot of literature which I didn't have time to go through. However, let me start here and say that we all know that science of nutrition is an art. By the way, I was a nutritionist. I was trained as a biochemist in Baroda, where nutrition was how we were taught biochemistry. And after doing my master's, I wanted to become a student of G.P. Talwar. He doesn't know me, he doesn't remember perhaps. I came and he made me stand there for 3 hours. He said, yes, stand on, I'll come back. And he never turned back. And I had to rush out and go back to Baroda. I did my PhD in microbiology there, and so therefore lost the privilege of being a student as back as in late 60s. Well, in my opinion, and I believe it very much, is that just only being an artist, knowing the art of nutrition is not enough unless and until you know enough of microbiology. And not that because I'm a microbiologist, but what has been said so far and what is going to be said in next 2 days will show that microbiology is most important and central to what all you are going to talk about. And by the way, I don't know how many of you know that 50% of the carbon on this planet is attributed to microorganisms. I was shocked when once I, was told about this, and I had to look back and see, is it really true? And it's because we don't even see these microorganisms which contribute to 50%. Okay, start with the wisdom. Well, I say Chinese medicine, but there are plenty of such things in our own medicine as well, Ayurveda and Unani system of medicine. They say if you had a choice of choosing between good digestive system and good food, you will choose certainly good digestive system. And it also says what is absorbed may not be, may be more important than what is eaten. Well, there we get the concept of bioavailability. And from there, we got an idea of bioenhancers. By the way, this term was coined in my institute where I come from, in Jammu, first time. And today, again, like what Dr. Sibbal was saying, if you go to Google and say bioenhancers, you will see hundreds and thousands of hits. Because this science evolved in the last 30 years. And one of the examples I have just shown here is bioprene, which is nothing but piperine. piperine was isolated, it's isolated from black pepper, and black pepper is, taken by everybody. I think Professor Ganguly said that, was trying to make a distinction between food and probiotics or prebiotics. Perhaps there are a lot of things in common when we are talking of traditional knowledge, traditional medicine, traditional products and probiotics, prebiotics. You will see there's a lot of commonality between these, and perhaps the combination has not been tried so far. I believe, although prebiotics are some where we are actually working very closely, deriving these from the traditional knowledge. Bioprene is, I think, almost about $100 million business today. It's nothing but isolated pepper, piperine from pepper, and this enhances the permeability. This enhances the transport of things from the gut, and also based on piperine. Well, I don't want to digress for this particular topic, but I would say this molecule has been used along with some of the antibiotics in our previous work, and we have plenty of evidence, in fact, clinical evidence that the pharmacokinetics of some of the drugs can be altered in favor of using far less quantities of antibiotics, getting almost twice as much influence or effect, or what is called as Cmax, using the small quantities of piperine. I'll stop it here as far as the traditional knowledge is concerned on piperine. Well, I just try to put together some of the things in this format, and where gut microbiota will be, of course, discussed by everybody in next 2 days. That's what I believe, and a lot of focus will be on pre and probiotics, but immunomodulation as the focus, which already Professor Ganguly mentioned. But what I shall be talking about is some of my own personal experiences, as well as the strategies that we could build from this subject. Well, Professor Ganguly said that it may not be true that we start with a sterile gastrointestinal tract, but there is a lot of evidence, well, as the metagenomics is now evolving and being used more and more, perhaps we will know whether the gut is actually sterile, because in the initial stages the numbers will be so small that you may not pick them up. And we also know that majority of the microorganisms of the gut cannot be cultured, and therefore we have to focus on metagenomics. But in the process of the development, I was reading some of the papers that were given to me that I think its first 10 days are very crucial for a newborn child, and perhaps you start from 100 microorganisms to 10 raised to power 8 microorganisms, the total biome within 10 days, first 10 days. That shows that how important it is for exposure of the infants to right kind of environment and right kind of, and which cannot be anything better than mother's lap. And we have enough evidence to show that during the course of perhaps first 1 year, that the total microbiome of gut is evolved. And of course it's questionable when we talk about it, that is it dynamic or is it Is it same for everybody, every human being? It cannot be so, of course. There are lots of evidences to show that there is epigenetic component involved into this subject, and there are a lot of other things, which to which we are exposed, or the child gets exposed during the course of time. But there is a component of evolving internally and finally coming to a point of what we say is 100 trillion microorganisms that make the gut microorganisms. And 100 trillion, it's sometimes mind-boggling. We can't even count the zeros, 10 raised to power 16, when we are talking of 100 trillion. And, well, it is said that if we put them all in one sequence, 2.5 times it will circle the entire planet, and that of one single human being. By the way, we have microorganisms not only in humans and animals, like what we are talking today about, but we have huge population of microorganisms in plant kingdom, what we call as phytomes, what we call as, I mean, they are very, very important as far as the secondary metabolites are concerned. And so therefore, the importance of these microorganisms is what is leading us to looking for the right ones and using them as the probiotics. Now, which one to be picked up? Which one to be selected selectively, so that we can we can decide. That's okay. Because I'm not able to see from here, so therefore I thought I have a printed version of it. I would rather like to look from here. And what is important is operational taxonomic units, which they say is almost about 1,000. Lot of things are actually presumptions, because we also know that only 20% of the microorganisms that we have in the gut are actually culturable. And rest of them we can now reach and understand only through metagenomics. And a lot of work has been done, which I had an occasion to look at, and people have gone to various geographies and various kind of people, and they have shown that it is not exactly the same. However, there is some kind of a uniformity of about 50%, where you can see the establishment of these microorganisms in the gut. But there are large number of activities, physiological activities of the human beings, which are attributed to probiotics, or which are attributed to the gut microorganisms, and selectively few of them have been worked out, and that is what is practically making us to look at probiotics as a formal, or what's called as agents that could be used for the health promotion of the human beings. Now, here this cartoon gives us 4 locations where you can see the concentration of these microorganisms. You can see here that the major concentration of this is the colon, and you can see the colon has practically almost 10 raised to power 8 or 10 raised to power 12 microorganisms. That means 1 trillion microorganisms will be found out of 100 trillion in the colon itself. So our gut microbiota can be pictured as a microbial organ. If you take this whole thing as one, you can call it a microbial organ placed within an organ. That's the power of gut microbiota. So I have already talked about it. 100 trillion friends you did not know you had. And of course, there will be some foes as well, because they keep on fighting, and there will be definitely some which will be there. But Professor Ganguly mentioned that most of these are our friends. He already talked about the person who coined the word probiotics, and I don't have to elaborate on this, but he proposed that the acid-producing bacteria in fermented milk products could prevent fouling in the large intestine, and if consumed regularly leads to longer, healthier life. Well, we have, just a couple of days back, we saw a result of one of the very large clinical study, about 25,000 subjects, which was done in England, and they have shown that a majority of the people who were put on yogurt for almost 11 years, follow-up, and they found that 28% of them could be, they found the diabetes reduced in the 28% of them. I mean, they were the subjects who were selectively taken, who had tendency to develop diabetes. So what are the gut microbiota? I'm sure most of the microbiologists sitting here know them. Predominant, almost 80% of the gut bacteria belong to these four phyla. And you can see some of the genera that I have taken out of this, and these are very well known. In fact, what I was working upon in my lab almost about 10 years was Bifidobacterium and Lactobacilli, because we were using these practically for producing enzymes, industrial enzymes, and producing also lactic acid, which was used for polylactic acid fibers. And the role of these bacteria in the gut system, particularly for anti-inflammatory activities, was actually worked out in IIIM Jammu by a couple of my students. So I just to show a couple of them here, Bifidobacterium was actually where we focused upon. And I think I remember about 45 years back, this was my first encounter with microbiology, was at Alembic Chemicals, Sarabhai Chemicals in Gujarat, in Baroda, where we were working on lactic acid production. And I started working on Bifidobacterium and Lactobacilli, casei, and others at that point of time. So extensively used live organisms, which is what probiotics are some of them are mentioned here, and it also includes Lactobacilli casei, which is used by Yakult extensively. And you can see some of these Lactobacilli brevi, Lactobacillus acidophilus. These are the organisms which various companies are using as probiotics like drugs. And but there are a large number of companies which are making them in large quantities, even in India, in fermentation plants, and then freeze drying and preparing preparations which are exported out to various parts of the world. There are some other examples which are generally not known to many people. For example, this pediococcus is one which a lot of people have actually used for lactic acid production, and it's a homo-fermentative organism which is unique, and this is also being used as probiotic. But one of the very important organisms which not many people talk about is yeast probiotics, and particularly Boulardii is one, and Saccharomyces boulardii, which some of the people in India are actually extensively using for animal, probiotics, and this is also used as a host for expression of some of the enzymes. And in my first slide, I forgot to tell you, I had shown a cartoon which was about Aspergillus niger. That's why I spent half of my life working upon, and Saccharomyces boulardii and Aspergillus niger are two which are used as hosts for heterologous expression of the genes, which where you can get up to about 200 grams of a protein expressed in 1 liter, and therefore they are very, very important organisms besides being used as probiotics. I think Professor Nirmal Ganguly also mentioned about the immune system, and we said that gut is the major site for immunology, immune modulations. I think here is what is a commonality that I can see with traditional medicine and herbal drugs and probiotics. We have seen in our research for almost 15 years that large number of products from traditional medicine cannot be actually traced through pharmacokinetics in the streams. Even if you give them in gram quantities, we do not find them either we thought that they are not actually getting assimilated, or what is happening. Practically, they just pass through. But during the course of that retention time in the intestine, they do their job. And this is what is happening practically with the probiotics. I think there is a commonality here with respect to some of the, I would say, elite products from Ayurveda, like we call them Trikatu, Triphala, and if you see the components of these, you can never trace them through pharmacokinetics, even if you use the latest state-of-art technology where you can go to PPT levels, but we fail to find some of the vital molecules that actually are responsible for the activity. But this is the same story with probiotics. You cannot trace perhaps some of the very small quantities of the products that they generate, and then these are the ones which actually act at the gut level. But I'm reminded of a product, I think I talked to Nirja when she visited me, again, a Japanese product which I had the privilege of working upon. It's almost $1 billion product in the market today, and most of the women know it more than men know. It's called SK2. This is a fermentation product, and it was used as a drink in Japan, and Procter & Gamble picked it up, and bought the whole concept, and today it's a $1 billion product for what is called a skin, what's called regeneration, skin regeneration. And in fact, we had the privilege to work on it scientifically for the first time. Even Japanese company in Kobe, where they were producing this in large quantities for the entire world, had not actually worked into the mechanism of action of this, although it is clear that it was one of the best products on the market. It's called SK2. And we were surprised to see that there were very small molecules. We had to use MALDI-TOF to actually reach those molecules which are generated by interaction of the microorganisms. By the way, large number of the lactobacilli which were actually being used inadvertently. They did not know actually what kind of fermentation is taking place. But this was traditionally for almost about 200 years being used in Japan. And today it's the property of Procter & Gamble. And so I see a lot of correlation between these small molecules which are produced by these friendly microorganisms, and one of the major outcome of that research was that we found that they were mostly anti-inflammatory agents. And as Professor Ganguly said, that inflammation is central to anything that we are talking about today. And we even showed that the beauty product for women, for what is called as face lifting, was actually anti-inflammatory natural products being produced by microorganisms. I have enumerated some of the important functions that we know now through the research of these colonizing microbes, and most of us know some of these, and have been characterized completely, like small chain fatty acids, vitamins, controlling the pH, which is very, very vital actually, and so on and so forth. But one of the most important, exciting information which I could gather is how the probiotics absorb some of the chemical ballast that is produced or that is consumed by the humans, and they absorb it on them, modify perhaps sometimes. This is where we were involved in finding the enzymes, specific enzymes from this microbiota. Well, a lot of work was done on termite gut or on rumen bacteria as far as the metagenomics was concerned, to look for specific enzymes. I think the best, research that I could see of industrial importance was finding a cellulase from termite gut. And you can imagine a termite, and we could never ever cultivate the microorganisms, but we went through the route of what's called metagenomics, and today the best cellulase available on market is from the termite gut. So you can see there are some critical elements which are now extensively known through the literature, which is attributed to colonizing microorganisms. We also know by and large, the gross activities. How do they do it? We can see that there is a transient colonization, which is very important actually, because if you did not have this dynamic mechanism there, then perhaps the fouling will take place, then perhaps it would be very, very difficult for this total microbiome to be so active. So therefore it's a transient process. And immunomodulation, as I said already, is one of the very important defensive mechanisms which is attributed largely to all food that we eat, and also to the probiotics and the organisms. I don't have to go into the details of how this immunomodulation takes place. This is a preliminary experimentation which is done on animals all over the world, and we are doing it almost every day, and you can see the inflammatory interleukins. You can always see upregulation and downregulation. There are in vitro technologies available with us, so we can always test these. So I think this IL-6 particularly, and IL-2, IL-10 has been extensively worked out, and also along with this cascade of apoptotic enzymes that have been worked out, which are reported. However, whether in the clinical setting these work and to what extent they work, how does it alter with the alterations of the microbiome is a moot question. And of course, you can see large number of structured clinical trials that have been taking place all over the world, but it needs definitely to be more focused so that one could, it's again like, finding a pin from the haystack, basically, because once you are talking about a trillion microorganisms doing something, which particular one and where you are actually focusing upon. Well, I like the idea of having only one microorganism or one lactobacillus and develop perhaps 10 raised to power 10 load of it and ballast it. Well, that's very safe. But whether or not it works or not, there are people who would like to mix 8 or 10 of them together and give this as probiotic because they're not very sure which one is going to actually work and which one is going to work with who gets the ballast. So I think these are issues which have to be actually, I'm sure they are going to be discussed here. Those are the actually things which are going to be taken forward in near future. Again, this is what I said about the macrophages, are responsible for the Th1, Th2, mode of controlling the inflammation. And, well, here again, I would just say, when we're talking about the single molecules from nature, again, I'm coming back to this, and I take the example of piperine. We worked on macrophages. We found that, Mycobacterium tuberculosis, bacilli, when actually we were using piperine, it actually inhibited the what is called as ABC transport system, and therefore that was the mechanism by which we could see that we can alter and modulate the macrophages. I'm sure some of the microorganisms, some of the products of these bacteria are also doing a job like this. One has to actually move forward to see this. So again, I would just say dysbiosis, and we are trying to use these microorganisms to bring back the dysregulations to the proper regulations, and would say it's quoted like I drink until I'm healthy. It's actually quoted from I drink until I pass out. And here actually you are trying to now see whether you can at very early stage you can start with probiotics. We need ample evidence, actually clinical evidence, to say at what point of time child should get exposed to these large amounts of microorganisms. Well, I already said about what is the role of probiotics. You can see I have prepared a list of what is already in the literature and what has had some evidence attached to it. So I think one has to actually pick up the right kind of approach, right kind of target, and then on the basis of that one could see that how the interaction takes place. I just showed that there are almost about 6 things through which we can decide where we should use the probiotics and in what format should we use. I have just found out a few recent publications, and in fact this first one, which is reduction of type 2 diabetes, I have just followed it very extensively, particularly with the background that in traditional Indian knowledge, I was told almost from almost 2,000 years, the yogurt was used very extensively by Vaidyas in this country, and they were actually using it through enemas. And if there is a problem through having it, pass through the gut, and this was practically used when we did not even know what is diabetes or what are other cancers and other things. So I think I would like to shorten my lecture by coming back to the use of probiotics. What are the challenges actually we have? I already enumerated some of the challenges, what will be, what we are facing almost when we are trying to use these probiotics. And say the dose, in fact, part of it was already reflected in Professor Ganguly's remarks. I think what I'm worried about is the formulations that we make. What should contain in these formulations. Of course, it has to be palatable. It has to be acceptable. It has to be acceptable to the children on whom you are focusing. But I think a lot of research is required to be done on this. We have to actually take into consideration the prebiotics. We have to take into consideration the additives that we are adding on to this, and I think that's a challenge which will, even though I am told that 300 million Yakult bottles, 6.5 billion per, no, you said the consumption all over the world, 30 million. Okay, 30 million bottles a day, which is quite large, but it should be 300 million. It should be in India alone, perhaps. So how is it going to be acceptable to the authorities that be? How is it going to be acceptable to the consumers? And how can we see that we are not actually adding on something which after 10 years we'll come to know that, well, we made a mistake. So I think those are the issues from the regulatory point of view we have to look into. I would not like to delve into the legislation, which I'm sure it's very already important for your company, your foundation to look into, but this concept is becoming increasingly popular. Well, we have data on that. And they say with consumers, because of the heightened awareness, so that is what we are trying to create. I'm sure this is a forum, if the proceedings of this are made, I think in the past they have done an excellent job. I got some of them which I was looking into, but if you look at the scenario of global consumption pattern which is coming, I think in 2018 they are showing up to 2018 consistent, perhaps more than 5 to 7% increase in consumption. If you see, the Asia Pacific is coming up very large numbers, and rest of the world is also trying to pick up of this. What is most important is what you are going to do here, and what the foundation is going to do in next couple of years is this scientific substantiation. In fact, any incremental substantiation that you can do through your research will become very, very important. That's where we have to focus upon. Now I've said what I had to say about probiotics, like we saw Professor Sibal talking about his, what's called as institutions. I just thought I'll give 2 slides on Jamia Hamdard, which, by the way, Jamia means university, and we are using university twice. When we say Jamia Hamdard University, it is wrong. It is Jamia Hamdard, it means Hamdard University. Well, you can see on the left, Pandit Jawaharlal Nehru, who actually helped to conceive this idea of, at that point of time, it was in 1962, just a few months before he passed away, he was responsible for the idea maturation on Indian Institute of History of Medicine that was built at that point of time with the philanthropic contribution of Hakim Abdul Hamid, who was a practicing, traditional Hakim. And in 1989, Rajiv Gandhi actually, opened the university as an autonomous university, only about 15 minutes drive from here. And today it has become an excellent center for research, but we are focused on healthcare. And we have all the modern elements of education and research. And one of the most important points which I thought I should bring in here is the last point here, on-campus setup of international labs. We have Becton and Dickinson, which is very well known for the flow cytometry. We have a center which is an all India research center on our campus. We have Hillman Laboratory for vaccine development and research, and we have Ranbaxy Laboratory for clinical research on our campus. And we have a couple of more. I would say, I would give credit for at least two of these to Nirmal Ganguly, who when I moved here from CSIR, actually was responsible for to make these people to move to our institute. And today, these are the centers which are doing excellent job and helping Indian students, educational institutions, and the practicing medical fraternity, for example, in the form of teaching flow cytometry by Becton and Dickinson, Hillman Labs for the US FDA approved Center for Research in Vaccinology. And I have already discussed with Professor G.P. Talwar, and he is very keen and interested in this. Only the day before yesterday, he was talking to me about it. And clinical research, we have almost for last 20 years on our campus. And the new hospital setting, which is coming up, has one floor dedicated for translational research, which is going to actually of course, our focus is on traditional medicine, but we are not behind as far as testing probiotics and other products that could come our way. So I think the conclusion is that we should see it as a crucial requirement for securing the future of probiotics as a functional food ingredients. That's part of it. I try to make it within the time that has been given to me. I was told that I have to talk about 25 to 30 minutes. I might have crossed, because I'm not using a watch, so I would have perhaps got disturbed, but thank you very much for your attention, and thank you again, Nirja, for inviting me to be here this afternoon. Thanks a lot.
Thank you, Professor Kazi, for that wonderful insight on the importance of the gut microbiota and the role of probiotics in favorable modulation and the way ahead. May I request Professor Ganguly to kindly give a small token of appreciation to Professor Kazi.
Dr. Kazi once gave me a shawl.
In Jammu.
Jammu, it was a Pashmina shawl.
May I request Professor Ganguly to kindly give a small token of appreciation to Professor Nair. And may I again request Professor Ganguly to kindly give a small token of appreciation to Professor Anupam Sibal. May I request Professor G.P. Talwar to kindly give a small token of our gratitude to Professor Nirmal Kumar Ganguly.
It should at least
Yes. Thank you, Professor Kazi, Professor Takeda, Professor Ganguly, Professor Nair, and Professor Sibyl. We now move on to our scientific session. We now have the keynote lecture, and may I request Professor Ramach.
सारी निकाल दे एक बार में एक लगेगा मैनुअल लगेगा ना 20 मिनट लगाने में लगा रखे हैं वो भी कॉपी कर ले चुका है सारी एक वो तो कॉपी दे दी मैंने सारी दे दी सारी जा रही है मगर ये वाला इनका पेन ड्राइव शो नहीं कर रहा ना दिक्कत है हिडन हो गई क्या हिडन नहीं पेन ड्राइव शो नहीं कर रहा पता नहीं क्यों नहीं आ रहा किसी और में लेना पड़ेगा लगता है किसी और पेन ड्राइव में लेनी पड़ेगी किसी और पेन ड्राइव में लेनी पड़ेगी हाँ ले नहीं रहा ये पेन ड्राइव शो दिखाई नहीं रहा क्यों नहीं कर रहा हिडन में तो नहीं चली गई कुछ है इसमें नहीं ये तो ऐसे है कुछ नहीं है इनकी पेन ड्राइव शो नहीं कर रही कुछ होगा ही नहीं इसका मतलब कॉपी किया है नहीं तोड़ नहीं निकल जाती है निकल जाती है उसने कह रहा हूँ गिलास तोड़ दे किसी और पेन ड्राइव में लेनी पड़ेगी इनकी लेके आएंगे कुछ डाटा इसमें है इसका लैपटॉप में किसमें डाल दी चल रही यहाँ से कॉपी चल रही हो गई कहाँ डाल दी कॉमन फोल्डर में अच्छा
We now move on to our first keynote lecture. May I request Professor Rama Chaudhary and Professor Anupam Sibbal to kindly chair the keynote lecture. This keynote lecture was to be delivered by Professor A.K. Srivastav, Director and Vice Chancellor, National Dairy Research Institute, Karnal, but because of some unforeseen circumstances, he could not come today. So, Professor R.K. Malik, Professor and Head, Dairy Division of Microbiology, NDR, National Dairy Research Institute, Karnal, has been very kind to deliver the talk on his behalf. Can we request the chairperson to please introduce Professor Malik?
Good afternoon again. On behalf of Dr. Rama Chaudhary and myself, I'd like to extend a very warm welcome to our speaker, Dr. Ravinder Kumar Malik. He's a microbiologist by training, and he's Professor and Head of Dairy Microbiology Division at the institute that has done some amazing work in the area of dairy research and has really been a shining light of what can be done from some of these research institutes. He's based at Karnal. Very extensive list of publications, 110. Lots of chapters and manuals, and he has 3 patents to his credit, has been the recipient of many fellowships and awards. He is also the executive editor of the Indian Journal of Dairy Science, and a member of several distinguished academic bodies. So ladies and gentlemen, let's put our hands together for Dr. Malik, who's going to talk about dairy foods as a carrier matrix for probiotics.
This one? Dairy, niches, yeah.
Good afternoon, ladies and gentlemen. Honorable Chair, Dr. Rama Chaudhary, Dr. Anupam Sibbal, distinguished, very senior professors, scientists, medical persons, ladies and gentlemen. As already informed to you that this lecture was to be delivered by Professor Dr. A.K. Shrivastava, Director and Vice Chancellor, NDRI, but due to certain pressing reasons, he just couldn't make it. So I have been assigned this duty to present this talk before you. In fact, I would like to thank, first of all, Professor Anupam for introducing me and Professor Rama for chairing this session. Well, the focus of my talk is on the role of dairy products or the dairy foods as a carrier matrix for probiotic organisms. I was just sitting over there and thinking that, looking into the importance of dairy foods as a carrier matrix for probiotics, why not the title of this particular symposium should have been Probiotics, Microbiome, Dairy Foods and Gut Health. Because at the very beginning, when Professor Ganguly was giving his inaugural address, he mentioned about the dairy products time and again. And then I could really think that, well, the talk that I would be giving would be very appropriate, as he's, as a head of the family, is giving a lot of importance to the dairy products. Now, coming to this presentation, of course it is well known that our gut is inhabited by several trillions of organisms, and of course this whole of this microbiota is very complex and heterogeneous, and we have got more than 1,500 species and 7,000 strains present over there in the gut. So this is an enormous one, and the composition is of course individual specific and serves as a fingerprint. This has been recently found, and in totality, these microorganisms in the gut, they play major role in the modulation of the gut function. Now, there is another question that comes out, that is, can the microbiota communicate or talk to the brain? Yes. Gut has a mind of its own. 100 million neurons in the gut. That is more than what we have in the spinal cord. And these neurons in the gut and in the brain, they are in constant communication with each other. And this is what the researchers have begun to explore, that there is a crosstalk of biomarkers through which microbiota and host communicate. And the recent research also shows that the probiotic bacteria can help reduce the anxiety and elevate the mood. That is the recent research, that how by taking the probiotic foods we can have elevated mood, or that our anxiety can be reduced. So that is one important factor, that our anxiety should be less. Now, as far as the probiotics in dairy foods are concerned, they are complement to each other. I would say that the probiotics and the dairy foods, they have got, in Hindi they call it a choli daman ka saath, as far as the gut health and well-being is concerned. And as far as the market, of course, there is a great demand the market for these probiotic dairy foods is going up, not only in India, where it's growing at a good rate, but in the Western countries too, I have seen there is a great demand. You go to the shelves of the food stores, almost 90% of the products, which I see that around 20-25 years ago, they were just simple dairy products. Today they are the probiotic dairy products. And people in those countries, I have seen that they have got awakening. They think about their health because the cost of the going to hospital is very, very high. So they have taken the health in their own hands, and they feel that by consuming the probiotic dairy foods, probably they can have better health. So that realization is giving a boost as far as the market of the probiotic dairy foods is concerned, which I think in India, we too have to realize this particular fact that the probiotic dairy foods are very healthy and we need to consume them. We need not go to a doctor for our smaller problems. So the probiotic dairy foods can take care of all those things, and of course that we can say that with a lot of confidence that dairy products would dominate the functional food sales in the coming years. And now coming to the history. Our forefathers, our ancestors, they used to take a lot of dairy foods, and particularly the fermented dairy products. For example, dahi, lassi, or such allied fermented products. And they had longevity. I myself, when I look back, when I think about these probiotic dairy foods, fermented dairy products, I sometimes wonder, and now I think, earlier I was finding that why my grandfather or my great-grandfathers, they survived for around 100 years or maybe more than 100 years. What was the reason? Didn't they have tensions? Probably they too had the tension that we have today. And in spite of the fact that they didn't have much of medical care during those years, they didn't have that much money to spend on themselves, but still they had a long life. What could have been the reason? The reason exclusively was that they had been consuming a lot of dairy products, a lot of fermented dairy products, and that gave longevity. And now when I study in the books about the Mechnikov and other such great personalities, then I try to correlate the two. Yes, that could be true. And here today I find that that's what we are advocating today, that fermented dairy products have long been considered an ideal vehicle for delivering probiotic bacteria. At that time, they didn't know what were probiotics and what were other organisms. But we can now realize that some of the strains that were being used for the fermentation of dairy products, they must have been definitely probiotic organisms. Today we have isolated those organisms from those fermented dairy products, and we have made it as a class of probiotic bacteria. And now, dairy foods and probiotics, they are a perfect combination, in the sense that they are an excellent food matrix for optimal expression of probiotic functionality. So this is the point that needs to be seen, that needs to be investigated. How come the dairy products are an excellent matrix as far as expression of probiotic functionality is concerned? This is being proved today, as on today, that we at NDRI are working on that particular aspect. How dairy foods are better than other foods as far as the expression of the functionality of these probiotic organisms is concerned. Some important genes encoding probiotic functions are induced on exposure to milk. Milk have got certain properties that help them as far as the expression of those genes is concerned, and they possess inherent properties to buffer acid in the stomach and increase the survival of probiotic organisms in the gut. I don't know, many people would be knowing that milk products, or the milk itself, is a very good buffer. And since the stomach is an acidic environment, we need to protect these probiotic organisms over there. So what can be a better medium or better matrix as far as the carrier of these probiotic organisms is concerned, when they would go along with the milk products to the stomach, there would be a environment which will not be that harsh as far as the survival of these organisms is concerned. So the consumers are having no positive perception about the dairy products because they are the integral part of our diet and is considered as healthy. Everybody knows that milk products are healthy. So this is taught even to a small child since beginning that yes, you must take milk because it is healthy. You must take dairy products because they are healthy. So they are a part and parcel of our life. And so there is an awareness that live bacteria used in the fermented dairy foods are also helpful, and consumers now prefer functional ingredients. As I said, that there is a awareness among the people that we must take the functional ingredients and they should be in the, could be a better medium other than the milk and milk products. So these are some of the examples of the probiotic dairy foods that we have today on the food market, that is in the yogurt or dahi, kefir, cheese, powdered milk, ice cream and frozen products. Chocolate is also, the chocolates are having probiotic organisms, and we are going to have butter that would be having probiotic organisms. So now coming to the milk and cultured dairy products, they are the storehouse of bioactive ingredients. So besides probiotic organisms, we have to also look into the bioactive ingredients that are present over there in the milk, because there could be a very good combination of the two. Many bacteria are unable to survive the acidic environment. As I already said, the dairy products provide that buffering medium, such as milk, yogurt, cheese. They can buffer the stomach. Then now, as far as another factor, the technical factor that comes into being is the refrigeration that keeps probiotic bacteria stable and viable. Yes, these dairy products, they are kept at refrigeration temperature. So definitely, when the dairy products are being kept at refrigeration temperature, the organisms are going to survive better. Then next is the cultured dairy products are a complete healthy package. Yes, it's a well-known that dairy food contains a unique combination of nutrients that work together to improve the health. Consumers now perceive health benefits not only from the calcium, protein, or the vitamin D that a cultured dairy products can offer, but also from the naturally friendly bacteria. So that is the current realization. So now what are the advantages of delivering the probiotics through dairy foods? The components that are present in the dairy foods, they have got a synergistic effects on probiotics. That is the current research that we are going to have, how the bioactive peptides that are released when the probiotic bacteria grow in milk, they act in combination with the probiotic organisms and provide the benefit. They have got a synergistic relationship. They have formed a synergistic relationship. And as a study that has been conducted in New York that showed that lactoferrin in particular enhances the bifidobacteria multiplication. So that is the beauty that the milk contains lactoferrin and we can grow the bifidobacteria. Otherwise, you know that the bifidobacteria is a very difficult organism otherwise to cultivate or to multiply. And the dairy products have also been associated with building strong bones. More recently, they have been implicated they have been shown to have they have got a role as far as the weight management is concerned. And now, as you know, that nobody wants to grow fat, take up weight. So that is a good combination. You take the fermented dairy products, you control your weight. Beside managing your weight, you carry the good organisms. And now researchers are identifying the factors which affect the gene expression. As I said in the beginning, that gene expression is better in the dairy products when we consume dairy products. So now that research is going on, that what are those factors which affect the gene expression of probiotics in dairy foods and also enhance the probiotic survival and activity. So these things are going in combination. And in the years to come, you will see that it would be proved scientifically that, of course, dairy foods have got certain components, and those components would be identified that how they help in the expression of genes of these probiotic organisms. Now coming to the synergistic effect of some of the components that are formed or that are present in the milk, as far as their relationship with the probiotic organisms is concerned. So lactulose is one such that acts as a bifidus factor. It is formed from lactose during heating of milk. If you look at the chemistry of that heating of milk, some part of the lactose gets converted into lactulose, and everybody knows that lactulose has got anti-constipation properties. It's because alkali isomerization process is used otherwise for commercial production of lactulose, and this lactulose is utilized by bifidobacteria for their growth and multiplication. And as on today, there are certain infant formulas where lactulose is added specifically. Then there are other synergistic effects such as they control the chronic constipation, as I said. They also control the salmonella colonization. They inhibit dehydroxylation of primary bile acids, exert antitoxin and anticarcinogenic effect. They also protect against DNA damage. Now, there is another component, that's the oligosaccharides, that's the GOS, galacto-oligosaccharides. They are also synergistic to probiotics. There are certain novel pharmacological functions that have been attributed to GOS as a prebiotic. They help in the proliferation of bifidobacteria that activate the cell wall and extracellular immune system. Also, they have been implicated or they have been used for cancer prevention. That is, the GOS helps in the suppressing the activity of enzymes that convert pro-carcinogens to carcinogens. And also, there are many more such good properties of these galacto-oligosaccharides. Then, there are certain bioactive peptides which are formed when the certain proteolytic enzymes are released from those lactic acid bacteria of those probiotic organisms, and then they degrade the casein and other whey proteins present over there in the milk. Those bioactive peptides, they are very, very synergistic to the probiotic organisms. So that is a recent topic of research. Earlier we had been talking exclusively about the probiotic organisms, what role they can play in the gut. But the current thinking is that why not include those bioactive peptides too along with the probiotics? Because those bioactive peptides, they have got almost similar type of health promoting properties as that of the probiotic organisms. And it has been found now that there is synergism between the two. That's why the fermented dairy products, as a carrier of probiotic organisms, they are not just 1 and 1, 2. That is the bioactive components present in milk and the probiotics. But they are 1 and 1, they are the 11. So that is the beauty, that's what makes these fermented probiotic dairy products as the foods which are not only carrier of probiotic organisms, but also they have got multiple health effects because the different bioactive peptides such as opioids, antihypertensive, antibiotics, and the prebiotic effect, and the probiotic effect, and the antithrombotic properties, immunomodulating properties, and other mineral binding properties, they are put together. These 7, 8, or 9 different properties of the bioactive peptides put together along with the probiotic organisms. I think they are doing the wonders, and that makes say these fermented dairy products as one of the important not only carriers of those probiotic organisms, but also put them at a higher pedestal. Now, there are other health promoting effects of these fermented probiotic dairy products, because this focus is exclusively on the fermented dairy products or the dairy foods. So they are directly through interaction with the probiotics. That's indirectly through microbial metabolites that are generated through the fermentation process. That is the biogenic effect. So these biogenic metabolites, that includes vitamins, proteins, peptides, oligosaccharides, organic acids, and also the fatty acids. Now, looking into the pharmacological and therapeutic effects of probiotic dairy foods. They are helpful in the diarrhea. I hope everybody knows about it, particularly in the antibiotic-associated diarrhea. Helicobacter pylori, it has been shown that in this particular infection, there have been certain trials have been conducted recently. There are randomized trials on H. pylori eradication. The efficacy of fermented milk-based product was found to be better than the capsule or sachet-based probiotics for eradicating intestinal H. pylori. So that is the beauty that the probiotics in fermented dairy products form, they have got a better role to play. Similarly, in the respiratory infections, some such studies have been conducted, and it has been found that the combination of algae, bifidobacteria, L. acidophilus, S. thermophilus, given in the fermented milk pod form that reduce the nasal colonization and the potential pathogenic bacteria in the healthy adults. So there have been several such studies and other such studies as far as the pharmacological and therapeutic effects are concerned. There's a hypocholesтерemic effect. Now it has been shown through certain cultures when they are propagated in the milk, they decrease the total cholesterol and the LDL, and they could increase the HDL. So that's what we want. Other such pharmacological and therapeutic effects include allergies, cancer, immunosenescence. So this is important, that is, in elderly patients when the immune system is going down, it's important that the daily consumption of probiotic cheese containing Lactobacillus rhamnosus, certain strains have been identified, and Lactobacillus acidophilus such as NCFM strain that has enhanced the parameters of immunity in elderly volunteers. So these are some of the novel researches that show that these probiotic organisms in the dairy foods, they could help in different aspects, in different ways, in different populations. Of course, there have been several other studies as far as the probiotics in the fermented dairy products are concerned. They have improved the gut mucosal immune system in malnourished mice. These studies have been conducted in small animals, and they have given very good results. Similarly, oral administration of supernutrients of milk fermented with Lactobacillus helveticus strain that enhance the gut mucosal immunity by improving the mechanisms that reinforce the epithelial and non-specific barriers. And similarly, they have been able to, fermented milk products have enhanced the intestinal mucosal immunity, mediated IgA cells and by cytokine production. Other such studies have been probiotic dahi. This study was conducted at NDRI Karnal, where it was shown that this particular strain in the fermented dahi, in the form of probiotic dahi, that improved the mice immune system and protected against sublular enteritis. Similarly, there have been other such studies which have shown that they are helpful in combating the different infections, different salmonella infections or other such gastrointestinal problems. And also now, another novel aspect that has come up in the recent years, that is the induction of satiety and the weight management by probiotic dairy foods. At NDRI also we are working on this particular, our group, one of our group is working on this particular aspect, where we are looking into how that these certain strains of probiotic bacteria in the fermented dairy products, they stimulate these gut hormones, GLP-1, GIP, CKPY, etc., etc. And there have been such very positive results as far as the small animal studies are concerned. And also these probiotic dairy products for cardiovascular health and diabetes, type 2 diabetes. I hope this is everybody in this hall would have seen this particular report or particular papers. They must have seen that Cardiviva is one such product that is having Lactobacillus reuteri strain that has been validated clinically, probably in Canada. And this is the world's first disease-specific probiotic, and it maintains cholesterol levels that are within the normal range. And Glucoviva that shows the promising for treating type 2 diabetes. At NDRI also we are working on this how the probiotic organisms, probiotic fermented dairy products can help in combating the type 2 diabetes. And of course, again, when we talk about the diabetes, weight management, obesity is and the point that comes, because the obesity comes first and then comes the type 2 diabetes. So there have been a lot many studies as far as the use of fermented dairy products in weight management, in managing the obesity, and then the type 2 diabetes are concerned. So there have been certain evidence-based recommendations for probiotics in different dairy products. For example, prevention of Clostridium difficile associated diarrhea in adults. You need to have a fermented product, and the number of cells is 10 is to the 10 twice daily. And similarly, as an adjuvant therapy for H. pylori eradication, again you need to have a certain number of cells in the form of fermented dairy products. And then elevation of some symptoms of IBS, again you need to have fermented dairy products fermented with certain strains of probiotic organisms, and you need to take those products twice daily, so as to have that, as per the definition also of the probiotics, that you need to have certain number of organisms. That is important. So there have been such evidence-based studies. Now, what are the, just 2 minutes, what are the issues on functionality of probiotic dairy products? Now, this is the forum where we have to look into the issues, where we have to discuss these issues so that we could come out with a paper, we could come out with certain recommendations that how we can further, say, take up the case, or how we can make certain recommendations to the government, and how what could be the way forward in this regard. So there have been studies using fermented milk products that have shown positive or synergistic effects, suggesting that probiotics are more active in fermented milk than in the capsule form. So this is, I think, this has set to rest everything, that definitely fermented dairy products are a better option instead of the capsules. But the comparative studies assessing the differences of the efficacy of the same strain in 2 different metrics are lacking. We need to carry out those studies, and there is also another point, another issue that we need to discuss, we need to understand, is the need for personalized probiotics. That's the effect of probiotics, not only determined by the probiotic strain, but also by the host's genetics, gut flora, diet, as well as lifestyle. So thus, it necessitates that need for a personalized probiotics. So that is another issue that we need to take up. And then there are certain technological issues that need to be addressed. That is interaction of probiotics with starter organisms. We need to carry out extensive studies, because probiotics itself in the fermented products may not serve as a good, they may not form a good fermented dairy product. So they always need to be, the product need to be always fermented with another starter culture. So what could be the compatibility of those starter cultures and the probiotic organisms, so that both grow simultaneously? There is no antagonistic effect of one on the other. And what are those starter cultures which are very, very compatible with some of the proven strains? So these are the issues that need to be addressed, as far as this is the technological aspect. And the probiotic viability, as you know, is very, very important as far as the processing stages and the entire shelf life of delivery for effective dosage is concerned. And then, again, we need to understand the damage or injury that is caused during the freeze drying or on thawing. We need to have those resilient strains of lactic acid bacteria, or the probiotic lactic acid bacteria, that can withstand even the higher temperatures. We need to develop that technology. Say, it could be microencapsulation. Because what I understand from a group in New Zealand at Reddit Institute of the Massey University, they have perfected a technology where the probiotic organisms can survive at 25 degrees Celsius. That is, we need not refrigerate it. So that is the question today. When food is a matrix, or dairy products is a matrix for carrier of these organisms, then temperature is also another important aspect. So that's what we need to understand, that how we can make these probiotic organisms resilient to this sort of environment, that the climate that we have in India, the tropical climate. Maybe we need to develop such microencapsulation techniques so that the organisms survive even at 37 degrees Celsius, and then comes the packaging. So these are the important issues. Now, these are the key issues before the Indian probiotic industry. We have got a limited consumer size. It is, half a minute, just in the cosmopolitan cities like Delhi, Chandigarh or Bangalore, people know about probiotics. How about the smaller cities, B class, C class, D class cities? So this is an important point that we need to bring about the awareness of the fermented dairy products in particular, and the probiotic fermented dairy products along with that, so that the people get to know and they consume these probiotic fermented foods. And again, there is limited accessibility due to high cost. We need to reduce the costs. Well, that's a question for the industry to think about, how it could be made available to the masses in such a good form and at a much cheaper rate. So we need to boost the consumer confidence by producing high quality, safe and cost effective probiotic dairy foods of proven efficacy. And these are the future perspectives, and the way forward, that is the probiotic milk products, they are expected to top the list of the probiotic cultures. I think 5 years down the line we will find that these are the only probiotic dairy foods which are say dominating the market as far as the functional dairy products is concerned. And we need to have special probiotic dairy foods for specific medical conditions such as gastrointestinal disorders, diabetes, obesity, hypertension, cancer and allergies. When we have got such a wide variety of these probiotic fermented foods, I think the market would automatically be there, the customer would be there, because the people from the different groups, different segments with different problems would come to the market for picking up those particular disease-specific probiotic dairy foods. And to conclude, that dairy-based foods, including ethnic fermented products, are promising vehicle as far as the probiotic delivery is concerned, and clinically validated probiotic dairy products will provide a benchmark as far as the boost, so as to boost the consumer confidence in these products. And finally, the probiotic dairy foods can bring health and wellness. Hence, motto should be to bring good gut health through probiotic dairy foods to as many people as possible, to build this nation, this India, a healthy nation, and to finally to say that probiotic dairy foods will provide safe and natural approach to combat diseases. It will be the medicine in the 21st century as we used to have antibiotics in the 20th century. Thank you so much.
Thank you, Dr. Malik, very nicely elaborating on the role of dairy food products. I think one of the very important thing is it is acceptability. I think the Indian, culture has that acceptability to dairy products, and anything given in the form of dairy products is easily accepted by the population. And especially we already know the health benefits of all the dairy products which we consume. It has been our culture. I remember our mothers used to give us curd before leaving for any exam or any interview or anything to alleviate the stress. So I think those things which have been inculcated in Indians is very easy approach through these dairy products to add probiotic strains of whatever type or the strain required, whether they need to be tailor-made and given to the population, and whether it is diarrhea, diabetes, lifestyle diseases. I think it may be the answer to all the problems. And I really thank Dr. Malik for elaborating the science and the chemistry behind it, which many of us didn't know, and all the biochemical, biomedical, bioactive peptides which are involved. Thank you very much for your elaborate presentation. Thank you very much.
Thank you, Chair, and thank you, Yakult Foundation and Dr. Neja for giving me this opportunity. Thank you very much, everybody.
We just request Professor Rama Chaudhary to just give a token of appreciation to Professor Malik. Professor Rama Chaudhary is the Professor, Department of Microbiology, All India Institute of Medical Sciences.
Okay.
Anyway, let us And may I request Mr. Yoshirao Nakamura from Yakult Hansha Company Limited to kindly give a small token to Professor Rama Chaudhary. Good. Thank you, Professor Rama Chaudhary. Thank you, Professor Sibbal. Thank you, Dr. Malik. We are running a little late, so I'll just request the chairpersons to adhere to the time, please. And we move on to our next session. I now request Professor B.S. Ramakrishna and Professor Shinjini Bhatnagar to chair the session, Gut Microbiota and its Modulation. Professor B.S. Ramakrishna is the head, Department of Gastroenterology, SRM Institutes for Medical Sciences, Chennai. He is the editor, Indian Journal of Gastroenterology. He has received several national and international fellowships. He is a recipient of several awards and has more than 140 publications in peer-reviewed journals and 20 book chapters. Professor Shinjini Bhatnagar is Dean Clinical Research and Professor and Head, Pediatric Biology Center, Translational Health Science and Technology Institute, Gurgaon. She has received several national and international fellowships and awards, and she has several publications in peer-reviewed journals. Can I request the chairpersons to introduce the speakers, Dr. Bala Murugan Ramadas, Dr. Jayashree Paul, and Dr. Johan von Bleich.
Thank you, Nita. We'll begin the session without wasting further time. I'd request Dr. Ramadas Balamurugan to please come on stage. Dr. Balamurugan is an assistant professor at the Wellcome Research Laboratory at the Division of GI Sciences at Christian Medical College, Vellore. He's been awarded several scholarships and fellowships. He's also a member of the National Academy of Medical Sciences and of the National Academy of Sciences. He has several awards to his credit. So, welcome, and over to you.
Thank you, Dr. Shinjini Bhatnagar. I would like to thank the organizers and my mentor, Professor B.S. Ramakrishna, for this opportunity. In the next 20 minutes, in the next 20 minutes, let me try and explore the influence of diet and infection on gut microbiota. Our body is dominated by microbes, and most of these are bacteria. The human gut is host to trillions of bacteria, and they are rich in diversity. Their membership varies from person to person, and it varies across the body sites. They are most fairly permanent, and they are resilient to changes. By now, I think we know why we care about normal flora. These common cells perform variety of protective, structural, metabolic, and physiological processes. It was rightly described in the past as forgotten organ, mostly because of lack of technology. And now, with advent of multi-omics, it is now possible to fairly, more reliably study the microbiome. The metagenomics has allowed us to understand the genetic potential of microbiome and to also know who are present. And metatranscriptomics, metabonomics, and metaproteomics has allowed us to understand what they do. However, culture and qPCR techniques are still crucial to understand more mechanistic and targeted questions. However, the association between microbiome and a host relationship is difficult to understand, or it is more complex one, because it has been implicated in almost everything from restless leg syndrome to cardiovascular disease. And there is an increase in accumulation of results on more correlative studies, and there are very fewer cause or effect studies. This can be mostly attributed to the better understanding of gut microbiome, which is in the last 3-4 years most possible. To do this, it is important to understand the gut microbial composition in health, and to understand its function and its implication on, or its impact on other microbes, and its impact on the host itself to maintain homeostasis, in maintaining the homeostasis. There are various factors in health that determine this balance. They are age, the socioeconomic status, gender, geography, host physiology, lifestyle, and host genetics are few to mention. Diet and infection are the two components that I'll quickly go over with sharing my experience and results from our laboratory. Before getting into details on the effects of diet, it is important to look at the food map of India. It is evident that across the country we are also diverse in terms of the dietary habits. We are highly diverse in terms of dietary habits across the country, and the standalone components that I can think of as the spice and homemade fermented food that are different when compared to the Western world. So the dietary habits are of two different components, the long-term and the short-term. The long-term component majorly determines the core of a human gut microbiome. Armugam et al. In Nature published that there are 3 different enterotypes that are present, and these are defined depending on the type of long-term diet habits that they practice across continents. However, short-term dietary habits also rapidly and reproducibly known to change the microbial pattern. However, they don't disturb the core. This is a recent publication by David et al. To share more of my experience, a newborn is sterile, and when it is expected to be sterile, and when it is born, the first bacterial inoculum that a baby gets is dependent on the mode of delivery, and the first feed that a baby gets is the breast milk. So we profile the microbiome using the NGS, the 16S based 454 sequencing, and then we found that there is low actinobacteria and high firmicutes that are found in formula-fed infants when compared to breastfeeding and breastfeeding plus formula-fed children. However, it was not influenced by mode of delivery. When we found that babies born with a cesarean section had low bacteroides and low proteobacteria when compared to other delivery modes. To answer slightly a different question, we chose a more homogeneous group in terms of diet, socioeconomy, and geography. And then the people were differing only in terms of age. We selected children above 3 years and till the adult agehood, and then we profiled bacteria using quantitative PCRs. These were targeted bacterial profiling, and then we found that Lactobacillus acidophilus, Eubacterium rectale, and Faecalibacterium prosnexi groups followed a trend where they stabilized in the middle age and then they dropped down in concentration. And this was differently seen. This was different in Bacteroides, where they kept increasing in concentration. However, Bifidobacterium stayed fairly stable across age. So there was this long-term, but this group had a similar dietary pattern, but they all differed in age. So we went back to two groups who belonged to the same age group, but they differed in their long-term dietary pattern. So the red is of more of a low socio-economic dietary pattern, whereas the green is of a group of people who are more of animal-based diet and they don't consume milk and milk products. So what we found is that these two groups have two distinct signatures. They're two distinct cores. So this is evident that long-term diet can determine the core. To show a snapshot of the representative bacteria of those two cores, we find that the Faecalibacterium predominates in the group which consumes more of animal-based diet and no milk and milk products, while Lactobacilli totally is disappeared from that group. And we also found that Bifidobacterium is also absent in the same population, On the other hand, we have infection which transiently perturbs gut microbiome. So we found that gut microbiome alters in acute diarrhea and in infection. So in a study where in children with below 3 years old, and then when they had diarrhea, we collected samples at 3 time points, where the first sample was diarrheal sample, second was the first form stool, and then third was 3 months later. And we found that the bacterial profile was different in the first and the second time point, while it came back to supposedly normalcy in the third time point. What attracted us in that result is that we found that there is increase in Lactobacillus acidophilus group in the diarrheal samples. So we speculated that that this has got to do something with the dietary intervention or the traditional dietary intervention that the people followed. Also, in an asymptomatic condition, we found that the the gut microbiota did not change. So here in this study, we screened neonates for rotavirus infection, and all those neonates with positive for rotavirus were profiled for microbiome, and we found that there was no change. And another infection condition where in children below 3 years, and another parasitic infection where we found that in children below 5 years, we found that the red is the infected group, where green is pre-infected group. We found that the Prevotella group increased, while the Lactobacillus also increased in the infected group as observed earlier using real-time PCR, whereas here we used the NGS using 16S targeting 454 sequencing. We again compared it with post-infection. We found that the post-infected group again went back to like the pre-infected group. Here we had 3 time point sample, where the first sample was a pre-infected group because this was an ongoing study for various for a different reason, while the third sample was a post-infected. So we found that Lactobacilli was nowhere there in the first and the third sample, while it increased during infection. So considering all the results that I've showed, from a sterile newborn, microbiome undergoes succession based on various influencers, including diet, infection, medication, and then they show a trend to increase in diversity as they grow older, and then the diversity peaks at adulthood, and then it drops back. So believing that the diversity provides healthful benefits. To conclude, we feed the microbes they take care of the diversity, they determine the enterotype, and then these in turn provide beneficial, helpful function to us. By studying baseline in our country will allow us to derive signatures in health and disease, and that might help us determine personalized dietary intervention. Thank you.
Thank you, Dr. Balamurgan. I think that was, we'll go on with the next talk and we'll take questions as a group at the end of it. Thank you so much.
I'd like to welcome Dr. Jayashree Paul. She's at the she's FLU Foundation Fellowship, MB Fellowship felicitated by the Vice Chancellor of Andhra University, Visakhapatnam, and has been honored at the 90th Science Session of Indian Science Congress. And currently she's working as a scientist and has several publications in her area of work. Dr. Paul.
Okay, good afternoon, everybody. And I am very thankful to the Yakult Foundation and the supporters, THSTI and Apollo Hospital for giving me an opportunity to share with you my research experience with my research team. And I am also grateful to the chairperson, Professor Ramakrishna, and Professor Shinjani Bhatnagar to chair this session. And I am going to take you from the dietary habits that has been talked to, fermented food that has been talked to. I am taking you to a different story of comparative assessment of the predominating gut flora during inflammatory bowel disease. Now, as we know that this inflammatory bowel disease is an autoimmune disease, and there is no pathogenic organism has been found so far to be associated with the disease. And it is a multifactorial disease. It has a genetic component, and it also has a microbial component which exaggerates the immune response. So far this is the story that we know about the inflammatory bowel disease, which involves two diseases, ulcerative colitis and Crohn's disease. And the study that we have done in our laboratory mainly concentrates with the ulcerative colitis patients, which we have collected the samples from the gastroenterology department of All India Institute of Medical Science with my collaborator, Dr. Vineet Ahuja. Now, coming to the contributions of the microbiota to the host, which has been deliberated in detail in the morning session as well as in the afternoon session. And what we see that the digestion and the fermentation of the carbohydrates are is very important contribution that the microbiota makes, and it also involves in the production of vitamins, development of the GALT, that is the gut-associated lymphoid tissue, polarization of the gut-specific immune responses, and the prevention and colonization by pathogens. And in turn, the gut immune responses that are induced by commensal populations regulate the composition of the microbiota to maintain the homeostasis. Now, a paper which appeared in Nature Review Immunology in 2013 says that the gut plays a protective as well as a pathogenic role. Now, in a normal individual, we find that the gut microbiota is in a symbiotic relationship, and the types of bacteria that are present so far, of course, the genome projects have come and microbiome projects have come, but still there are many bacteria which still remains unclassified. So there are certain unclassified bacteria, there are certain beneficial bacteria, and there are certain pathobiomes. Now, the purpose of these pathobiomes are that the useful bacteria keep the pathobiomes population down. so that the gall development and the Th17 cells, Th1 cells, which are responsible for the production of the pro and anti-inflammatory cytokines, can take place, and it also helps in the barrier function, REC3 generations, etc. And it also plays an important role in the production of Treg cells and the anti-inflammatory IL-10 cytokine. And these symbionts also keep, in the due course, they keep the pathobionts in control. But when a dysbiosis takes place, or when there is a disease and there is an alteration in the gut flora, it seems that the dysbiosis, because of the dysbiosis, there could be a production of the pathobionts, which are not coming from outside. They are indigenous flora. But some of these pathobionts might dominate during the disease condition. And because of this, it disrupts the microbial community structure, and there is an accumulation of colitigenic pathobionts. The pathogenesis of IBD is mainly located in, if you look at the pathogenesis, we find that it is located in the areas with high bacterial concentration. As I told you, there are two diseases. In case of Crohn's disease, it is mostly located in the terminal ileum and cecum, and in case of ulcerative colitis, it's the rectum area. The central event in the pathogenesis of IBD is the exaggerated immune response to the commensal intestinal bacteria or to its specific members. And this idea came because in a genetically modified animal model of IBD, do not develop colitis under germ-free conditions. So this clearly shows that there is a huge role of the microflora or the indigenous microflora in the manifestation of this disease. Now, we started our work somewhere around 2008, and that time we are working on the mucosal flora of the biopsy samples that we took from the hospital. And our results had shown clear delineation of the concentration of the mucosal flora between the predominating and subdominating genera under disease conditions. And in the previous speaker has already pointed out which are the dominant and subdominant flora in the gut, indicating that the subsets of bacteria participating in the pathogenesis of ulcerative colitis and Crohn's disease are likely to be different. And in the present study, what I am going to present today is to show, to evaluate and compare the changes in the diversity of fecal flora in the Indian patients. Right now we are working on the fecal flora of these patients from healthy to disease state during ulcerative colitis. And also quantified the bacterial population using certain specific probes that we have generated in our lab in control versus patient samples. And in the patient samples, we have taken three categories of samples: moderate, severe, and remission stages of the disease. And we tried to obtain any significant association of the bacterial flora during the disease condition. Now, these are the dominant and the subdominant bacteria of the human gut, which we have studied in our samples, that is Bacteroides, Bifidobacteria, Clostridium cocoides members, members of Clostridium leptum, Enterobacteriaceae members, Lactobacillus, Enterococcus, Atopoboium, Methanogens, and sulfate-reducing bacteria. And the techniques that we have used to illustrate these changes or the fluctuations in the gut flora involves fish microscopy, fish flow cytometry, and qPCR analysis. And we have also looked into the metabolites, the changes in the metabolites by GC analysis. And we have tried to do a preliminary study, this is still an incomplete study, to figure out the important changes that have taken place at the species level by using a denaturing page. And the probes that we have used for the flow cytometry are the EUV probe, that is positive control probe, for the to measure the total bacteria, which is leveled with FITC, and a non-UV 338, which is to see if there is any background signal, that is with non-UV 338, which should not amplify any of the bacterial species. And the genus specific probes that we have used were leveled with Psi5 at the 5' end. So this is the quantification of the Bacteroides prevetola group. Here we show the fish microscopy of whole cell in situ hybridization of the straight from the fecal sample. with genus-specific Cy5 leveled probe. So this is for the Bacteroides prevetola group, and down you can see the fish flow data which we have generated, and if you look at the graph, you can see that there is a control, there is a moderate, there is a severe, and the remission cases. And if you see carefully, you will see that there is a relationship when we compare the control, the severe and the remission. We see the fecal flora coming down, both by flow cytometry as well as the qPCR analysis of this Bacteroides flora. And this Bacteroides actually goes significantly high in the severe patients if we compare it with the control individuals. So the population of Bacteroides increases significantly during severe condition and reverts back to normal during remission. Now, when we quantified the genus Bifidobacteria, again with the flow cytometry, we find that there is a significant reduction which is expected in the Bifido population during the disease condition. But what we find that in the remission patients, again the flora comes back towards the normal level. And this was proved both by flow cytometry as well as the quantitative PCR analysis. And on the upper panel, we show the bacteria that are stained with the Cy5 leveled Bifidobacterium probe. So the population of the Bifidobacteria drops significantly during severe condition and reverts back during the remission stage. Coming to the Clostridium coccoides group, again we find that there is a significant drop in the moderate followed by the severe condition of the disease, and the patients which are in the remission stage again seems to revert back to the normal level of Clostridium coccoides, which is proved again by flow cytometry and the qPCR analysis. The members of Clostridium coccoides drop significantly during moderate and severe condition but reverts back during the remission stage. In case of Clostridium leptum, it was very interesting that we found that there was a drop of members of Clostridium leptum group, and there is a drop in the moderate as well as in the severe cases. But in the remission cases, we did not find a significant turnover or significant reversion back to the normal level, both by flow cytometry as well as by qPCR. So this shows that the members of the Clostridium leptum group, which actually comes down, they do not revert back so soon. It takes time probably. So the population of Clostridium drops significantly during disease condition but do not revert back during remission stage of the disease. So when we saw that there was a significant reduction in the butyrate producing bacteria, that is the members of the Clostridium group, we went little bit in detail to design primers and the probes for Faecalibacterium plasmodium, Roseveria intestinalis, and Eubacterium heli in particular. And we again quantified them using the fish flow and the qPCR analysis, which again showed that the members of Faecalibacterium plasmodium coming down in the severe condition and showing a tendency to come back at the remission stage, and so is with the Roseveria intestinalis, but Eubacterium heli we did not find a significant change. And this was true both by fish flow and the qPCR analysis. The decreased abundance of predominant butyrate producers like Roseveria intestinalis and Faecalibacterium plasmodium in disease condition was quite remarkable. And then comes the Lactobacillus enterococcus group. Now the members of Lactobacillus enterococcus group, what we find that in case of the moderate conditions as well as in the severe conditions, we find that there is an increase in the lactobacillus, which was a big surprise for us. So we are expecting that the lactobacilli population would come down, but maybe because of the dietary habits of Indian, population, the production of lactobacilli, even in the control samples, were higher when we compared with the literature in the European world. We find that the lactobacilli population actually goes up, and in the remission condition again we find that it is coming down to the normal flora. So this was something which was difficult for us to explain at this point of time. So the increased abundance in members of lactobacillus during disease condition was seen in our study. And these are the subdominant flora known as Atopobium cluster. And here also we find that even in the severe conditions, the population is low, but it goes up. So we don't know if all of them are pathobionts or not, but we find that there is an increase in the population of the Atopobium cluster during the disease condition. So there was a significant increase in the Atopobium during severe condition of UC. And in case of Disulfo vibrio, again we find that the population of Disulfo vibrio, that is the sulfate producing bacteria, are going high in case of moderate and severe conditions, whereas in case of remission they still maintain the level as we see in the severe condition of the disease. And this was seen both in the flow cytometry and the qPCR analysis. Now coming to the methanogens, this was very interesting that we found an increase in the Disulfo vibrio bacteria, but we found a decrease in the methanogens. The population of methanogens of course is low in a normal individual, but what we find that in the moderate and the severe conditions, the methanogens actually come down. And we specifically saw the quantitative analysis by PCR for the Methanobrevibacter smithii, and we find that in case of severe condition, we find a little high in the Methanobrevibacter species, but it was not significantly high. So the p-value was above 0.05. So there was a decrease in the methanogens during severe condition of UC. In this slide, what I show you is we made a comparative study of the firmicutes versus bacteroids. The members of the firmicutes versus bacteroids during control, moderate, severe and remission study. What we find that in case of moderate and the severe condition, especially in the severe condition, the ratio of firmicutes to bacteroids come down. So that means the bacteria's population go up, firmicutes go down. And during the remission, they sort of come back, revert back to the normal level. When we made a comparison of the gram-positive versus gram-negative group, what we find that the members of this ratio of gram-positive to gram-negative, that is, there is an increase. There is a decrease in the ratio of gram-positive to gram-negative because there is a change in the qPCR analysis. And in order to establish a probable link between the altered butyrate-producing bacteria and the impaired cellular metabolism, we also check the metabolites, that is the short-chain fatty acid production, using gas chromatogram profile of the fecal samples during these disease conditions. So what we did was, there is a standard, and we took the fecal samples and measured each of these metabolites. So there are, if we look into this figure, we find that there is a significant drop in the acetate production, in the propionate production, and in the butyrate production when we also measured the butyrate level in the fecal sample itself, which corroborated our observation where we saw that there was a drop in the butyrate producing bacteria. So among SCFA, butyrate and propionate concentration decreased significantly during the disease condition. And there was an increase in the lactate concentration. As I told you that we also found an increase in the lactic acid bacteria. So to prove this, what we did, can I take 2 minutes, please? So what we did was, we did a RICER study where we took a forward primer from the 16S ribosomal rRNA and a reverse primer from the 23S rRNA, and we wanted to amplify the variable region, and we took a genus-specific primer for, say, Lactobacillus in this example. I'll just show you one example here, where we have seen that there is a severe condition and the moderate and the control and the remission. We find that if we put it on a DNA chain page, we can see the number of bands which are different in these three conditions. So what we did, we took these bands and we sequenced them, and we went back to the database, and what we find that the population in the moderate, severe and remission, the bacteria which it shows matches are quite different. So similarly we did for the bacteroids, we did also for the, I'll just run through these slides, and we find that some of these bacteria are, for example, induces colitis in IL-10 deficient mice, this back one which was common in all category. So some of these bacteria could be connected with their functions as well. We did for the Clostridium leptum as well, Then we also did a phylogenetic tree of Clostridium leptum, showing the sequence homology.
Dr. Paul, you left.
Yeah, I'll just go through this. Let's run through this one, please. So this is a phylogenetic tree of 16S ribosomal RNA analysis of bifidobacteria, showing the sequence homology with the database. So what we concluded out of this, that since in case of the pathogenesis of the ulcerative colitis, inflammation is the central event. And what we found in our study is there is elevated levels of bacteroids which can exaggerate the mucosal antibody. And because it also, for example, Bacteroides fragilis is known to generate BFT or the enterotoxins. And in case of decreased level of butyrate producer was supported by the SCFA concentration or short-chain fatty acid concentration, which came down. There was an increased level of sulfate-reducing bacteria that is producing more of H2S, which is toxic to colonocytes and inhibits the butyrate utilization. And there was an increase in the lactobacillus, as I told you, and we found an increase in lactic acid production. There is a loss of, which can cause a loss of mucin layer, because we found a decreased expression of one of the mucin gene. So perhaps the adherence property of lactobacillus is lost because of the loss of the mucosal layer, and therefore it transits to the feces and gives us a high value of lactobacillus. This is our conclusion right now. So what we finally say that the dysbiosis can be functionally coordinated, because the dysbiosis in IBD are correspondingly not simply the structural changes in the gut microbiota, but are instead associated with major impairments in many fundamental microbial metabolic functions with potential impact on the host. So there is a reduction in all these clades may thus have a functional consequence on the ability of the host to repair the epithelium and to regulate the inflammation. So this is my research team, and there is a poster presented by Rina who did this work, which is displayed on the poster session. Thank you very much.
Thank you, Dr. Paul. Would you like to just sit here? We'll take questions later.
Oh, okay. And our apologies, I think the introduction was a little inadequate. So Dr. Paul is a professor, School of Life Sciences, JNU, and also a UGC research scientist. We'll go on to the third speaker, Dr. Johan van Likamama Vliet. My apologies for the pronunciation. He's an R&D director, gut microbiology and probiotics at the Danone Nutricia Research. So welcome. We'll be talking on the impact of food ingested microbes on commensal communities and host health. Welcome.
So thank you very much for this nice introduction. Congratulations with the pronunciation of my name. I think it was very good, actually. So I want also to thank the organizing committee and the highly established scientific community here that organized this meeting for inviting me. I am 2 meters and 4 tall, but I feel in fact very small in this order. So. You cannot hear me. Is it better now? I think I hear myself even. So what I will talk about is a sort of elaboration of some of the things that have already been introduced by others. And when I looked at the program, I tried to find some complementary entries to that. And what I will talk about basically is 3 things. First of all, I will elaborate a little bit more about where do these probiotic or food-ingested microbes, where do they come from? How did they end up in dairy, and how did they impact on our, let's say, our food production? And then, how do these organisms impact our gut microbial communities? And in the end, I would like to come to a sort of general model, which maybe can guide also future discussions on where fermented foods and probiotics can have an impact in maintaining health and establishing health. And the first thing I really want to stress is that working for a food company, we are really about prevention and keeping people healthy. It's not so much about targeting diseases. And that's really, I think, the objective of the pharma industries. But we are really at this left side of the curve. This is a curve that I took from quite an influential document that really shaped also, let's say, the European Union research politics about a decade ago. Now, let's go to food-ingested microbes. I will sometimes use the word food-ingested microbes, sometimes probiotics, because I think probiotics has also in legal terms a very heavy meaning. It needs to be linked to demonstrated health benefits. I think it's broader than that. So you will see the two terms appearing. And the general hypothesis for a long time was that, in fact, these microbes that we use to ferment dairy products, they originate from, grass, for instance, plant material. And there has been a moment that either via a cow, the milk production machinery of a cow, or maybe directly, they ended up into dairy, and then they evolved and optimized to fermenting dairy products or even other food products. And then what happens today is, in fact, by consuming such products, we continuously challenge our microbiota. And then we have, of course, different aspects of the microbiota. We have the upper GI tract, where these ingested microbes are temporarily overwhelming really the resident microbiota, and then in the colon they are still an important subdominant population. Now let's have a look. I found this paper in it's a Nature publication of a couple of years ago, and it's an archaeological study, and they found pottery in the Eurasia region, and look at the quote that I highlighted here. It says that the first evidence of, let's say, processed milk, and in this case it means fermented milk, dates back to 6,500 years before Christ. So that's when, dairying started. And then if we look at another, that's very quick. I will just continue. I think it's a mistake. So I looked at what does the word domesticate mean. Yeah? And if you look at the Webster's dictionary, it means to adapt an animal or plant to live in intimate association with or to the advantage of human. I think we can talk about our lactic acid bacteria actually as domesticated microbes, because this is what we do. So then key questions can be, how was this human-LAB partnership established? What were, that's the scientists' view, what were the molecular events of domestication, if you want, and can we reproduce that in the laboratory? And how can we expand our partnership and exploit lactic acid bacteria better to our benefit? And it's a study that I started before I joined Danone, actually, but it continued during my period at Danone. We worked on Lactococcus lactis, which is a major component of dairy starters and of the typical Dutch cheeses, and it has two important main natural niches. One is, my time is up again. I don't know which button to push. Okay, no problem. It has two important main niches, that's dairy and plant material. And what we did is we had genomes available of two phylogenetically closely related strains, but isolated from these different niches. And that's the KF147. You will see the strain code a couple of times, and the typical dairy isolate is IL1403. So what a PhD student, a very talented PhD student in my lab at that time did, his name was Herwig Bachmann, he in fact did a laboratory evolution experiment. And that's very simple. So what you do is you put the plant strain in milk and you let it grow in successive re-inoculations and see whether a better evolved strain appears. In fact, there was one trick needed. You also needed to introduce a plasmid, which is required for growth. And then he went on. So in the end, he did that for 1,000 generations, which took about 5 months. So there was a lot of patients involved as well. And then he isolated from each of these 3 independent experiments single colonies and did growth and acidification tests and looked at fitness. And so how well is a strain able to compete with another strain? And we did genome analysis, so resequencing and also transcriptome profiling, looking at gene activation patterns. So what were the results? Well, this is what you see if you look at the typical experiment. You see the KF147, which is the highest curve, and then after 300 generations it goes quicker, and after 600 even quicker, and after 1,000 generations you really have quick acidification. This is an acidification curve. We found something strange, which I will not elaborate on. If you have questions, I could really do that after the talk if needed. But we found that we lost, in fact, the protease, and that is explained in a separate publication which was published in ISME Journal 3 years ago. And then the single colony isolates, they more or less reproduce this. So we have the full curve, which is the original strain, the wild type. Then we had one strain which had some sort of intermediate acidification acceleration, and 2 that were really much quicker. So what we did then is we resequenced these genomes of these single isolates. And then what do you find? Well, you find a whole bunch of mutations. And I only present here the SNPs, and what you see is that these 4 individual strains, they had between these 3 individual mutants, they had between 4 and 21 mutations per clone. And interestingly, some of them were the same. So that really shows that independently you get the same mutation. That must be really a contributor to fitness. Now this is a very complicated slide, and I don't want you to understand it fully. I just want you to get the main message. So this is transcriptome profiles on milk. And the black dotted spot in the middle is the KF147, so the original strain on itself. Of every gene, you have the transcriptome mapped on itself. Then the IL594 is the dairy isolate. And what you see here is a straight line, and what you need to keep in mind is that the closer you are to the straight line of the next strains, the more close you are to a dairy-type expression pattern. And what did we see then? Well, if you put the original strain with the protease, you are very far off, because you're not in quadrant 2 or 3. And also this slowly growing strain is quite far off. But the 2 fastest growing strains, in fact, they seem to sort of tweak towards the dairy reference line, let's say it like that. We went further and looked at what type of things had changed in these transcriptome profiles. And what you see is that the adapted strains, they typically take up peptides, which is logical, because in milk you need to take up peptides to grow faster. And there was also something on branched-chain amino acids biosynthesis. Well, typical sugar uptake, other than lactose, was downregulated. So it really made sense in the sort of the ecological context. And we could trace that back to really the individual mutations. I will not show that, it's in the paper, but you can see that the mutations can really explain these differences in transcription patterns. So let's go back to this picture here, and now focus on the right panel of the slide. So how do these fermenting microbes challenge the gut microbiota? And what I will just want to do is to highlight something. We have already heard a lot about the gut microbiota, but I think we need to realize that from a sort of niche science area, it really has moved towards, I would say, a global and very, very important science domain. And for instance, the journal The Economist published on the cover in 2012 this as a main subject, and there's an extensive feature in this magazine. And also Science, the well-known scientific journal, chose it as the #1 scientific breakthrough area of 2013. So what do we really know? Because we see a lot of correlations, and here there's 2 slides, 2 pictures from 2 individual papers in Nature, and the left one depicts differences in the microbiome of obese and lean people. And I will not discuss it in detail, but I think what is interesting is that there are ecological models appearing. So we see that, for instance, short-chain fatty acid producers are down in obese people. We see that Enterobacteriaceae and Proteobacteria are going up. And we also see that global richness, so the diversity in the microbiome, is down in some of these obese patients. And then the second study did a dietary intervention, an energy-restricted high-protein diet. And what this study found is that, in fact, some people are less prone to respond to this diet. And that is related to the microbiota composition. And if people respond well, you can increase the richness, the diversity in the microbiota. So that's very interesting, but still, it helps to develop mechanistic models and testable hypotheses. I think it's very important for our science field. It also shows that diet is key, but we need to demonstrate causality. I think it's really, really important. Correlations only cannot be enough to tell the whole story. And this slide is a one-slide summary of a paper that we were involved in, which was published in Gastroenterology 2 years ago. And it's in fact a microbiota transplantation, a fecal transplant in people with metabolic syndrome. And it was consisted of 18 people, 18 obese volunteers with metabolic syndrome, and 9, so their colon was washed, and 9 of them, they received an obese microbiota. In fact, it was their own for ethical reasons, and 9 others received the microbiota of a lean donor. And the amazing finding was that, in fact, insulin sensitivity was largely restored in people that received the microbiota from a lean donor, showing really a causal component in the microbiota composition. And the microbiota analysis pointed again to this group of butyrate producers which we already heard about. So how can we really leverage this? I think I cannot stress enough that food is the main modulator of the microbiota. It was also already highlighted before. And then we had to talk about macroconstituents, microconstituents, prebiotics, and these food-ingested microbes. And of course, it's all in the context of epigenetics and lifestyle. In the last bit of my talk, I will talk about two specific models where we investigated this interaction further. So these models are, what we like to use is typically simplified models. That means healthy twin pairs, for instance, to eliminate some of the genetic diversity by human hosts, or dysbiosis conditions, typically in animal models. And the analysis is very much dependent on next generation sequencing technologies. So two examples, an impact of fermented milk product in healthy twins, which was published in 2011, and one in a collaboration with Harvard Medical School, a truck model of ulcerative colitis. So both studies, they used the same fermented milk product, which consists of 4 strains, in this specific case, Bifidobacterium, Lactococcus lactis, Lactobacillus delbrueckii, and Streptococcus thermophilus. So this is the first study. It's a healthy twin pair intervention. It was done in the laboratory of Jeff Gordon. And basically what these twin pairs had, they consumed for 7 weeks the product, and then there were a whole set of analysis done on these on fecal samples, which I will not elaborate too much on. But one of the key findings was that if you look at the fecal microbiota, there's no major perturbation due to the ingestion of these food-ingested microbes, which makes sense because they are, of course, relatively low numbers. So the composition was not significantly changed. Then, if you look at at one of the strains that was ingested, this Bifidobacterium strain, you see that if you try to do recovery from feces, you see that in this point of transient integration is nicely made. You see that only during the consumption of the product you can really recover the strain at significant amounts in fecal material. But there was something else which really surprised us. We also did transcriptome analysis on fecal material. And in fact what we found is that we could not only found a lot of transcripts of the Bifidobacterium, which was highly active, but we also saw that just the presence of this strain in both the human studies and in the parallel mouse model, we found that carbohydrate metabolism was really significantly regulated. And there are 3 examples here of individual enzymes in plant-derived carbohydrate metabolism which were differentially expressed and activated in this specific case, both in a mouse model and in a human model. And it's extensively explained in this McNulty et al. Paper. The last piece of information comes from a model of ulcerative colitis. A truck model which is developed by Wendy Garrett and Laurie Glimcher at Harvard Medical School, and it's a genetic model, and it's characterized by a heavy, spontaneous, and a highly penetrant colitis, which can be treated by antibiotics. So by wiping out certain microbes, you can really treat or cure the disease, and it's transmissible. So if you co-house animals, truck animals with healthy animals, the healthy animals will also develop the colitis phenotype. And what the group did before we started to collaborate with them is that they nicely identified that certain proteobacteria are in fact part of the cause. So you can isolate especially Klebsiella pneumoniae and Proteus mirabilis from cholitogenic animals. And then if you, in healthy animals, you put back these microbes, they will develop the truck. If you put these microbes alone, it's not sufficient for developing colitis. So they are really in concert with the endogenous microbiota causal agents. And then what we also did in a collaboration with them, we found, in fact, when we did 16S-based microbiota profiling, we found that one of the differences between the cholitogenic mice and the non-cholitogenic is that there's different levels of bifidobacteria. So upon colitis, you lose a large part of the bifidobacterial population, and that was confirmed by various methods. And then what we did is we did an intervention study with this fermented milk product, and we found that it ameliorates colitis. And what you see here is on the left, so you see here colitis scores. On the left, you see the fermented milk product, then you see a non-fermented control, which is in nutritional composition the same, and then you see also a sham feeding control. You see that especially the fermented product has a very good performance here. And the other one shows, the next slide is a similar type of slide where we compare, in fact, irradiated product and product with live bacteria, and again you see that live bacteria do a much better job. Then we looked, as in the previous presentation, in fact, to what might be causal agents. And we hunted down really metabolites in the cecum. And one of the surprising findings, in fact, was that if you had fermented milk product, you had lower levels of lactate than in the control and in sham feeding experiments. This was sort of counterintuitive because we introduced lactic acid bacteria, so we expected more lactate. And and a similar profile also for pH. And then when we looked further also to other short-chain fatty acids, we came to, we found in fact that though lactate was lower, so the other short-chain fatty acids, propionic acid, butyric acid, and acetic acid, in fact were increased upon the treatment with the fermented milk product. And we could link that back to the higher presence of specific butyrate-producing species. And then with subsequent the in vitro experiments, we found that short-chain fatty acid and pH, which was modified, really created unfavorable conditions for these cholinergic microbes, Klebsiella pneumonia and Proteus mirabilis in vitro. And the last slide that I will show on the research data comes from a publication which just came out a week ago, and it's something about, I wanted to make the point, contrast food versus medication. So what this laboratory did, they had various methods to treat the colitis phenotype in these mice, and the fermented milk product is only one of them, but also anti-TNF-alpha or antibiotic administration have the same effect. And if you then transplant the microbiota from cured mice to germ-free mice, in fact, the key message is that with the gentamicin and anti-TNF-alpha mice, they are really cured permanently. So the microbiota has been changed permanently, and it can this cured phenotype can be transferred. Well, for the fermented milk product, this is not the case. It's transient, and that is reflected by the presence of the food ingested microbes in the feces of both the donor and the recipient. So here's my last slide. I would like to share with you a sort of general model of gut homeostasis and the prevention of disease, and where food interventions could play a role. And it's inspired on a picture, a figure published by De Vos and Newdorp in Nature last year. I think we can be in this homeostatic situation, and then due to infection or antibiotics or maybe diet or lifestyle, we slowly enter into a dysbiotic state. We lose some of the good microbes, if you want, and some of the bad ones, they go up. We maybe have impaired gut barrier function, which is very often seen in early dysbiosis, and there's always this component of low-grade inflammation. And if you don't do anything, it will go on and we'll get tissue destruction in the end, and that will lead to disease and maybe even chronic disease. So what you can do is, of course, you can go in with pharma, and you can maybe do gut microbiota transfer, and then try to reverse this process and ultimately get back to homeostasis. But I think where food should play a role is in this early stage, when things are about to go wrong. Then we come in with our functional or nutritional foods or specific probiotics to sort of close the loop and make sure that we go back again. And I think that should be one of our models, and this also helps us to think about functionalities that we want to identify in our probiotics, like restoring barrier function or maybe anti-inflammatory, which we can select and develop further. And this is the last slide with the key conclusions. I told you something about domestication. I want to make the point that these food microbes, they transiently integrate and impact the commensal structure in some extent, but especially transcriptional activity and metabolite production, at least when we look at feces. And I think there's really a need for further translational approaches to fully harvest the microbiome lever in food and health. And the last slide is in fact the most important one, because these are the people that did the work. I thank you for your attention, and I hope it was not too long.
Thank you very much, Dr. Blake. Would you please take a seat?
Yes.
So we come to the end of the session. Essentially, we had three speakers. Dr. Balamurugan told us how the microbiota changed during life, and he suggested that all these changes were attributable to changes in the diet, which modulate them. Dr. Paul talked about changes in inflammatory bowel disease and showed that active disease was associated with changes in the microbiota, which reverted to normal or reverted towards the baseline when they recovered. And Dr. Lee actually essentially told us that the gut microbiota could be modulated by externally ingested microbiota, and that would change both the microbiome and the metabolic capacity. And it had, and it's really the host and the microbe both acting on each other's genes to induce something. So if we have time for two questions from the audience. Dr. Ganguly, please.
My question is that Balamurugan shows that change of microbiota with age, and you showed a crosstalk. Do we have data that, because some of the work which is coming up, that it helps in aging process, healthy aging process, the microbiomes. And so I would like to know from your basic work, both of you, what do you come across data set which shows that microbiota goes through some kind of a transformation during aging?
Is the question addressed to me, or? So, I can maybe give a start. So I think there are quite a couple of publications. I'm sure Colin Hill is... Does it work? Yes. I think there's quite some work going on that shows that in, let's say, in aging people, the microbiota is changed, and that correlates to immunosenescence or other adverse parameters. I'm quite sure that Colin Hill will talk about that tomorrow, because a lot of that work is going on in Cork, Ireland, actually. And yes, some of the signatures of dysbiosis, which I showed, low-grade inflammation, loss of certain beneficial bacteria like butyrate producers, is also seen there. So there might be, as with other benefits, good opportunities for probiotic or specific foods.
The only consideration is that there is this decrease in diversity in elderly population that we have observed. So any intervention that is possibly there to increase or even to elevate diversity would help.
One last question, Dr. Malik.
Yeah, my question is to Professor Paul. There is a confusion regarding the ratio of the formicutes to bacteroides. In literature, we find that in certain cases it is said that when the bacteroids are more, that is, the ratio is less, it is better for the gut health. Whereas in others, we find that the number of formicutes is more and the bacteroids is less, that is better. So we are a bit confused in that regard. What is correct statement?
Hello.
Thank you.
Now, the thing is that, are you talking about the disease individuals, or you are talking about the normal individuals?
No, related to obesity.
My study is restricted to inflammatory bowel disease, and where we have seen that there is a drop in the firmicutes, and there is an increased production of the bacteroids. So the ratio actually changes.
Yeah.
Okay, and of course, I mean, this is only at the genus level I am saying. In the species level, that's, we can actually figure out, that's what I am trying to do now, to figure out in the species level, in the firmicutes as well as in the bacteroids, if we can actually pinpoint which is the one which is going down and which is the one which is going up. But the ratio changes during the disease condition, not during the healthy condition. But regarding the obesity, I cannot make a statement right now.
Well, if you... I think many of us would be knowing about the papers from Kenny et al. And the Dilzane there, and if you read all those reviews, it's given that if the formicutes are...
Excuse me, Dr. Malik, I think we are talking about two different things. You're asking about obesity, and Dr. Paul's worked on inflammatory bowel disease. So I think we need to take this discussion outside during tea.
Okay, thank you.
I think we'll close the session. I'll hand this over to Dr. Neja.
Thank you, Professor Ramakrishna, Professor Shinchini Bhatnagar. Can we request you please to give a small token of appreciation, 2 minutes, quickly, to the speakers. And we can be asked Dr. Sarah Thompson and Mr. Richard Hall to please come and felicitate our chairpersons. Thank you so much. This was a wonderful session, but we are running a little late, and because of a paucity of time, can I request you to please be back here after tea and a good photograph by 4:45. We are still late, even after we come here at 4:45. So we'll try to... Thank you so much.
टिन्नी कहाँ से क्या कर दिया
you.